Good afternoon, everybody. I'm Shana Kelly. I'm the director of PRiME, and I'm really delighted that you join us today for this joint symposium between PRiME at the University of Toronto and UCSF. And this is a kickoff event for a new program at PRiME. It's the Connaught Global Challenges program, and I'll tell you a bit more about that in a few minutes. But we'll just kick things off today with a little bit of introduction and just a few housekeeping notes. So we will record today's symposium just to really make it accessible to all. There is live captioning, so if you need it, just hit the subtitle button or the live transcript function, and you should be able to see the captions. The chat function is disabled just because of the size of the group that we have. But you can certainly use the Q&A interface as we have speakers presenting. If you a question occurs to you that you want to ask at the end when we do the Q&A, just throw it right into that interface and we'll feed it over to the speakers. If you have any issues as things are going, you can email Angela from our team, and this is her email address. So I'll just pause for a second in case you want to make note of that. But that's all we have for housekeeping today. Okay, so we have a really terrific lineup of speakers today. I'm incredibly excited to hear from each one of them. And so the way we've laid this out, the topic for today is the chemical and structural biology of proteostasis. And we have some different perspectives coming in on that topic. Again, I think it's going to be a very interesting session. We're going to spend the first few minutes while people are still joining and getting settled in to tell you a little bit about PRiME and the Connaught Global Challenge program, in case this is your first time hearing about it. And then when we hear from Michelle Arkin, who's the co-organizer of this event, when we hear from her, she'll tell you a little bit about UCSF as well. But we'll kick things off. I'll just also mention our day 2 schedule, which is tomorrow. Same time, same place, and there will be shifting focus to talk about high throughput technologies to accelerate biology. These two areas were selected because there's really strength, we think, both at the University of Toronto and at UCSF. We have great investigators in these areas, and I'll tell you a little bit more about the program and how we envision these areas of strength really helping us build a strong partnership. And again, I just want to mention, in case I forget at the end, that we'll save the questions till the end, and then we're also going to have open discussion. If you have questions about PRiME, if you have questions about the Connaught program, questions about the science, obviously, or just the general topic, we would love to hear from you at the end. Okay, so I'll just give you a brief introduction to PRiME, tell you who we are, a little bit about our goals and some of our activities, and then I'll talk to you a little bit more about the Connaught Global Challenge program. So the University of Toronto, I'll just say a few words because there are a few people joining us from outside of Toronto and certainly folks from San Francisco. The University of Toronto is one of the largest research-intensive universities in North America. We have over 90,000 students spread across 3 campuses, one in downtown Toronto, 2 in outlying suburbs of Toronto, over 14,000 faculty, and a tremendous investment into research spending, specifically in biomedical research. So I honestly didn't know this until I moved here in 2006, but I think Toronto is really an epicenter for biomedical research, and there's a tremendous amount of activity and also increasingly a tremendous amount of impact. At the University of Toronto, we're very fortunate that we are right across the street from a major medical corridor. We have the Sick Kids Hospital focused on pediatrics. We have UHN, which actually includes a variety of different hospitals with different specialties. We have Mount Sinai, we have Sunnybrook up the street a little ways away from us. And so not only do we have this large research university, but we are nice and proximate to a great medical corridor. But we are big, and that's part of the reason behind why we decided to put PRiME together. So PRiME is what we call at the University of Toronto a strategic initiative. It's not a department, it's not a faculty, it doesn't teach undergraduates, it doesn't even teach graduate students. It really functions as a virtual research institute. And so what we've done is brought together really everyone across campus that is interested in biomedical research, disease biology, drug discovery, diagnostic development, to really bring together this community around research interests. And so we span 4 different faculties: medicine, pharmacy, arts and science, applied science and engineering, and 12 different academic departments. So our faculty base is very broad. We also have 125 trainees that are involved in PRiME. So we have a wonderful community that we're continuing to build. And the investigators that are involved in PRiME, these are highly collaborative and translation-oriented folks. And it's really been a pleasure to work with this group over the last year and a half that we've been getting off the ground. So our goals include elevating research in drug discovery, diagnostics, and disease biology, helping our investigators fund their efforts so that they can make progress faster and really accelerate the important research they're doing. We really try to empower collaborations, bring new investigators together for the first time to just keep new ideas flowing. We also have a training mandate, and we are really working to try to train multidisciplinary and translational leaders and also helping our trainees be equipped for the careers they may pursue in industry or other sectors. We try to create community in the large ecosystem that we have in Toronto. And we also are passionate about building a biotech ecosystem, having more activity, startups, larger entities, and trying to draw them into collaborations in Toronto for a variety of reasons, including so that we can build a strong ecosystem for our trainees to access once they leave us at the University of Toronto. So our research strengths, definitely on the basic side of basic science side of precision medicine, things like antibody engineering, all kinds of omics, functional genomics, nanomedicine and drug delivery systems, AI and drug discovery, target identification, precision diagnostics. We have a big contingent from our chemistry department, so lots of small molecule work, biology on a chip, high throughput screening platforms, and you'll hear about some of these areas kind of peppered through this symposium today and tomorrow. One of our flagship programs, and where most of our funding goes, is to our PRiME fellows. So these are individuals, graduate students and postdocs, who are working on new collaborations that bring at least 2 of our members together. And these projects are high risk, high reward. We seed them by funding the fellow and then try to build a fully funded project around that individual as the work takes off while they're a PRiME fellow. So this is our new cohort of fellows. We're very excited to see what they do over the next year. Lots of very exciting projects underway. I'll also mention, we do a variety of things within PRiME, but because we now have this nice self-organization around drug discovery and diagnostic development, when things like a pandemic happen, we're there, we're ready. We already have a group assembled that can bring their best ideas to bear on things like the COVID-19 pandemic. And so we have many people who have been working hard on new diagnostic approaches, new therapeutics, and we've been able to help them attract a significant amount of funding to their efforts. So now I will turn to the CNOT Global Challenge program, which is why we're here today. A little bit about the CNOT Fund. So this is a wonderful resource that we have at the University of Toronto, and it's got a very rich history. So basically, there was a lab that was established at the university in 1914 to produce the diphtheria antitoxin. Eventually, this is where insulin was discovered. So, incredibly historic. That's where Bansing and Best worked. The lab expanded, it produced insulin, and eventually it was sold. The university took that money that came from the sale. It's a bit of a sore topic with us because they sold it for a relatively small amount of money relative to what insulin has been worth to society as a whole. But in any event, the Connaught Fund was established based on that funding. And over the years, it has funded all kinds of collaborative efforts and commercialization readiness programs. And again, it's a tremendous resource. The Global Challenge program is to help the University of Toronto link to other universities so that we can, again, accelerate research and expand our reach, and also improve our visibility across the world. So we were successful in obtaining funding from this program. And so what this will do for us is it's going to link us with 4 top-tier universities across the world. So UCSF, Hebrew University, the ETH, and the National University of Singapore are the partners that we prioritize to be part of this program. So this program, the proposal was written pre-pandemic, so it did not contemplate the situation that we're in right now. What it did contemplate is having a speaker series where people from each of these institutions will come visit us at the University of Toronto. We also have a plan mapped out to have PhD students and also postdoctoral fellows exchange to these different universities to get experience in a different lab, learn different techniques that maybe we don't have access to at the University of Toronto. We are absolutely still going to do this, but obviously we need to wait until we get back on our feet again in terms of travel and hopefully conquering the pandemic. We also have a scholars program for undergraduate students to give them research opportunities. So I'm very excited about this. I'm very grateful to the Connaught program for giving us the opportunity. And given that we have this, I don't know, year of limbo where we can't necessarily get on planes and see each other in person, we thought, well, let's at least, get on Zoom and start really getting to know our partners, helping our partners get to know us, and really starting the conversation about how we can work together. And so that's what today is all about, bringing together some folks from the University of Toronto, UCSF, to really start exploring areas of mutual interest. So again, I welcome you. I'm glad you joined us. I want to introduce you to Michelle Arkin, who is, again, our co-host today from UCSF, and she is also our first speaker today. So we will move through this program, again, short talks and then Q&A at the end, general discussion. So just plop your questions and whatever points you want to have discussed into the Q&A interface. Let me go ahead and introduce Michelle, and then she'll tell you a little bit about UCSF, and then she will also tell you about her very exciting work. So Michelle is a professor of pharmaceutical chemistry at UCSF, and she is also now the department chair of pharmaceutical chemistry. And her lab, as you'll learn more about today, works on the chemical biology of novel targets, protein-protein interactions, mainly relevant for cancer and neurodegeneration. She's also the co-director of the Small Molecule Discovery Center, which is a collaboratory for high throughput screening, fragment-based drug discovery, and medicinal chemistry. Before she joined the faculty at UCSF, she was at Sunesis Pharmaceuticals. She was one of, I believe, the first employees of Sunesis, and she led their Department of Cell Biology and Drug Discovery and Cancer and Immunology. And one fun fact about Michelle, she was my lab mate. When we were both PhD students at Caltech, Michelle and I shared a lab. Everything I know about DNA conjugation chemistry, I learned it from Michelle. So we go way back, and I'm so delighted to have this chance to reconnect. So with that, Michelle, I will let you take it away. I'll stop sharing my screen, and you can share yours. And I look forward to hearing from you.
Thank you. Okay, great. So I apologize for not having a very nice green screen behind me. This is just my kitchen wall. It's a great pleasure to be here with you all today. And thanks so much to Shana for the introduction and for also including me in this exciting program. So a brief overview of UCSF. If I can get my thing to work. Okay, so UCSF, University of California, San Francisco, is a public university and a medical center. Medical center reaches, there are several hospitals in the city and then throughout California, especially Northern California. It's as a public university, it's the top public recipient of NIH funding for several years. And we have a terrific culture of collaboration, innovation, bioentrepreneurship. There are almost 200 companies that have been founded by UCSF faculty. And UCSF is really a temple to biomedical research, to the types of things that Shana was just talking about. It's everything we do is relevant to that. It's also a large university, and it's small, I guess, compared to Toronto, but we have 3,400 faculty, including many clinical faculty, about 25,000 staff. That includes about 3,200 postdocs, students, and fellows, more or less equally divided among those three. We don't have any undergraduates. It's a purely professional school. We have 4 professional schools in medicine, pharmacy, nursing, and dentistry, and then a graduate division. There are 19 PhD programs in the graduate division, all focused on fundamental biology, chemical biology, and biomedical sciences and clinical research. To help organize us, very similar to what Shana was describing with PRiME, we have this set of organized research units that help to facilitate interdisciplinary research, and they're organized around different diseases, and one that is cross-cutting and particularly relevant to today's conversation is the Institute for Quantitative Biosciences. So QBI is an institute that's really focused on biomedical interactions, biomedical research. We have a number of outreach programs very similar to what we're doing here today with PRiME, with exchanges, scientific and person exchanges between universities. So as Shana mentioned, that today we're starting with talking about protein homeostasis, and then tomorrow more of a synthetic systems biology kind of set of lectures. And it was very challenging to just come up with 2 topics to get started. There are so many areas of intersection between the 2 universities, and even within protein homeostasis, we have so many faculty that are really engaged in this area that I decided not to list them because I was sure I would forget some. But I send out the invitation that if you're interested in collaborating around protein homeostasis topics, feel free to contact me. If you have somebody in mind, I'll make an introduction, or if you don't, I'll be happy to recommend people. And I imagine Jason would also be happy to take your call. So today I'm going to talk about a project that overlaps with what we're talking about, the other speakers are talking about today, looking at a molecular machine called P97, and asking the question, if we have a multifunctional enzyme that's involved in many different types of biology, can we discover function-specific inhibitors or modulators, activators even, of multifunctional enzymes? And the reason we started thinking about this is because we're very interested in how to make drug discovery, chemical biology more precise and drug discovery more effective. And one of the big challenges is that biology is super complicated. So you can think about, say, a protein-protein interaction, but these 2 interactions really take place, these 2 proteins interact within a complex network. And these complex networks, here's a protein interaction network, can vary throughout a disease. So we're not just talking about a change in a protein in a disease, but those changes in proteins can cause a complete change in the protein interaction network. So this is an example of proteins that where the levels go up or down, or the interactions between proteins go up and down in cancer versus wild type cells. And you can imagine if you tried to take one of these proteins and knock it out or inhibit all of its functions, then you would really lose a lot of information about what makes this function particularly important or this function particularly important. And so we want to know, can we modulate the function of this enzyme or this protein just in the context of one of these interactions or a subset of its functions? So P97 or VCP is a good example of this kind of complex multifunctional protein. So VCP or P97 is a homohexameric protein in the AAA ATPase family, AAA family of ATPases. It's involved in protein homeostasis in several different ways. It's involved in removing proteins, ubiquitinated proteins, prepping them for protein degradation, unfolding them, and particularly proteins that are especially stable. Green fluorescent protein, for example, needs help from P97 to get unfolded. Also, proteins that are stuck in membranes, like in the mitochondria or the ER, proteins that are in strong protein-protein complexes, like in chromatin or aggregates. But it's also involved in a number of seemingly different activities in membrane remodeling. For example, Golgi assembly, autophagy, maturation of autophagosomes. And so we wonder, of these different activities, which ones are important to modulate in a disease, which ones are homeostatic and you want to leave alone, and how are these different functions regulated? So in the case of P97, at least part of the regulation of P97 function comes from a network of adapter proteins and cofactors that bind to P97, link it to specific substrates, specific subcellular localizations, and maybe even different functions. So we want to know, can we develop function-specific modulators? So one thing that helps us think about how we might develop function-specific modulators of proteins like P97 is how dynamic these proteins are. So here are 3 CryoEM structures solved in Sriram Subramaniam's lab of P97. We're iterating between these 3 different conformations of the protein that you see with different nucleotide bound states. The protein has 3 main domains: the protein-protein interaction domain in purple, an ATPase domain, the D1 ATPase domain in orange, and the D2 ATPase domain in blue. And so you see, as you turn over ATP, the protein-protein interaction domain changes conformation dramatically. And if you think about the central pore that goes down the center of this protein, these changes can also open and close that central pore. So Stacy Bulfer, a postdoc in the lab, was interested in how these different protein conformations may affect function. One of the reasons we're particularly interested in that, if you look at this structure, we highlight in red a residue that's at the intersection between the D1 domain and the N-terminal protein-protein interaction domain. This is the most common mutation in a set of mutations that cause a disease called multisystem proteinopathy, or VCP disease, or IBM-PFD, disease with many names, not many patients, but many names. So all of these mutations, almost all of them, map to the interface between the D1 and the N domain and alter protein conformation and function, which I think you'll hear more about later. So we're interested in what happens in this disease too. And we wonder, is this a disease of network rewiring? Because these mutations in P97 cause both gain of function and loss of function. So here in this histology slide, you see buildup of ubiquitinated proteins, but you also see proteins like mitofusin 1 that are important for mitochondrial fusion and fission are more rapidly degraded in these point mutations. So some of these point mutations activate the ATPase. So you may have some loss of function, some gain of function. And if you look at what proteins are pulled down by wild type P97 versus disease P97, you see that the protein network itself may be altered. So for example, if you look at wild type versus 2 of the disease mutations, you see this particular adapter protein, which is a ubiquitin ligase, is pulled down more in the wild type than in the disease mutation, whereas the opposite is true for these other adapter proteins. So Stacy asked whether you can see an effect of protein-protein interactions, the affinity of different protein complexes, based on the conformation of P97. And so she immobilized P97 on a Biacore surface, on an SPR surface, and measured binding of one of the adapter proteins, P47, to P97 in the absence of nucleotide or in the presence of ATP or ADP. In ATP, we expect the protein to be in this up conformation, in ADP in this down conformation. And you see, in fact, that the protein P47 does recognize the different conformations of P97. Here, by SPR, multiple doses of P47 across the bottom, you can see the degree of binding, and that P47 binds more tightly to the up state or the apostate, then to the ADP-bound down state. And these point mutations, which data suggests, stabilize this up conformation. In the disease mutations, you don't see this conformational coupling between nucleotide bound state and P47 binding. So suggesting that P47 binds to an up-like state better, and that up-like state is more accessible in the disease mutation. So you can see over several different protein-protein interactions over this whole network, if some proteins bind better to the up state, we see some proteins bind better to the down state. Other proteins don't recognize the up versus the down state, that this would rewire potentially this whole interaction network and alter different functions differently. So when we look at this protein, also this highly dynamic protein, we can see it as a set of gears. And there are many ways you could consider using small molecules to alter these gears. For example, you might inhibit ATP hydrolysis in either the top domain D1 or the bottom domain D2. You could do that competitively or allosterically. You could imagine trying to break the gears between conformations or manipulating the protein-protein interactions directly. So we've done several screens and different types of discovery approaches to find molecules that do different functions. And I'll just focus on a screen that Kenny Ang in the lab did, looking at ATPase function, really specifically trying to find allosteric inhibitors of ATPase activity. One of the best hits from the screen is this compound that we call the phenylindole series, because this turns out to be the core of the molecule. And this compounds went through a medicinal chemistry campaign with Donna Hearn's lab, Peter Whiff's lab at University of Pittsburgh, and also with some of our own chemists at UCSF. And together, we optimized this series of compounds. It turns out you could only make very small changes to the phenylindole portion of the molecule. You could make much more dramatic changes to this side chain, more peripheral part of the molecule. And an optimized compound here, which we call 245, has an IC50 for inhibiting ATPase activity of about 50 nanomolar. So how does this work? So it turns out that these compounds are allosteric uncompetitive inhibitors, meaning that they better to substrate bound state of the enzyme. We see that enzymologically by looking at the IC50, the apparent binding IC50 in the presence of different concentrations of ATP. So with a competitive inhibitor, the more substrate you add, the weaker the compound will appear because it's competing with that substrate. We, however, see the opposite. As you increase the concentration of ATP, we have apparently higher and higher affinity, better IC50s, the compound binding to that substrate bound state. So now to validate that concept, we looked again by surface plasmon resonance. So looking at direct binding assay, flow the compound over an immobilized P97 in the presence of no nucleotide, substrate, or product. We find that the compound does not bind at all really to the ATP, to the apo-bound state, the apostate. But when we have ATP or ADP, the compound binds much better, and it actually binds better to the product bound state than to the substrate bound state of P97. So to understand that better, we did some NMR experiments, which I don't have time to show, but also worked with Sriram's lab again to solve the CryoEM structures of the small molecule bound to P97. We did this in the presence of ADP because that was the highest affinity binding interaction. We arrived at this structure where you see the compound is bound to the D2 domain of P97, some distance away from ADP in the D2 domain, shown here in cyan. So how does this compound actually work? So if you look at a close-up of the structure, see that the compound fits in this, is highly complementary to this binding site in the ADP bound state. But if you put the compound into the ATP bound state, there are a number of clashes at this site. So this suggests to us that the compound acts as a wrench in the gears, keeping the protein from being able to cycle between the ADP and the ATP bound state, or inhibiting product release. So we can compare this allosteric inhibitor to inhibitors with different mechanisms of inhibition and see if they block different types of functions of P97, or if all the functions of P97 are blocked by all molecules. So we compared our uncompetitive inhibitor that doesn't bind to the unbound state, to a mostly competitive inhibitor from Nerviano and Genentech. This shows much more subtle changes as an effect of conformation or as an effect of nucleotide bound state. And one competitive inhibitor, this is a compound from Cleave Biosciences, a company started by Ray Deshays, and this is a clinical stage compound for cancer. And you see that it only binds to the apo state. It's inhibited by nucleotide. So Suifan Xu is our cell biology collaborator now at Caltech. And she looked at these three proteins, these three different mechanisms, and in some ways they're very similar to each other. They have a pretty similar level of inhibition of ATPase activity and a pretty similar level of cell killing in a cancer cell. But if you look in more detail, you start to see big differences in how functions are inhibited by these different compounds. So if you look at the Nerviano compound, for example, you see that it inhibits unfoldase activity. I'll describe this assay on the next slide, but it inhibits the proteasomal activity of cells, but it doesn't have much effect on autophagy or membrane remodeling. By contrast, our compound, the uncompetitive inhibitor, really has very little effect on ubiquitin-dependent unfolding, but it has a strong effect on autophagy. So here's an example of how that assay works. So the assay that we use to look at ubiquitin-dependent protein unfolding and then degradation is an assay where we have a GFP, green fluorescent protein, fused to ubiquitin. Then the cell or added ubiquitin ligases polyubiquitinate this protein, which then makes it a substrate for P97. It's unfolded through the central pore and loses fluorescence when it becomes unfolded. So we can measure over time fluorescence in cells, losing fluorescence. Inhibitors block that loss of fluorescence. Can do the same experiment in vitro. And these data are from Emily Blythe and Ray Deshays' lab. So here you see in the absence of P97, stable fluorescence. In green, the loss of fluorescence over time when you add cofactors and ATP. Compounds like CB5083, the competitive inhibitor, the non-competitive inhibitor, block that unfoldase activity, whereas our compounds don't block unfoldase activity, either in cells or in vitro. So we suggest that they don't bind to the conformation of the enzyme that's present in this unfolding state. However, they do have a strong effect on autophagy. So you see these yellow blobs that are evidence that we're forming more autophagosomes. That can either be because you're blocking autophagy or because you're increasing autophagic flux. And the data to suggest that these compounds increase autophagic flux comes from here. We see an increase in LC3, which is a key protein that sits on the membrane of an autophagosome, and that's in fact what we're looking at here. But we see an increase in degradation of an autophagy cargo protein, P62. So in other words, when you take a series of compounds with different mechanisms, you find that you can manipulate different subsets of function. And we're still trying to figure out why, but the uncompetitive inhibitors block or stimulate membrane remodeling activities, whereas the other series of compounds, competitive and non-competitive, block these ubiquitin-dependent unfoldase activities, P97. So in general, our goal is to use what we've learned about P97 conformation, develop function-specific modulators, use those to tell us which functions we want to be modulating in disease and which we want to leave alone for homeostasis. And from that, we can endeavor to do drug discovery for cancer and for these degenerative diseases based on knowing what it is that we're trying to modulate. So this is a big team effort. I want to thank Ray Deshays, who got us started down this path when he was at Caltech, the research team at UCSF, University of Pittsburgh, UCLA, and our fearless leaders at Leidos and NCI, and our collaborators in Sriram Subramaniam's lab for CryoEM structures. So just one more minute to talk more generally about what we do at UCSF and what we do in the Small Molecule Discovery Center. So we're really interested in a wide range of novel targets come from the community. And when we approach a new target, there are several different tools that we have at our disposal, depending on what we really think is going to work, what we know, what we don't know. So as with P97, we can try to take advantage of dynamics and allostery to address different mechanisms for inhibiting an enzyme. We also do fragment-based drug discovery and specialize in making covalent inhibitors. Sometimes that's the only way to get high affinity. We also do a lot of phenotypic screening. So if we understand the cellular context and maybe not what the ideal target is or the ideal context for that target isolated, we can use a phenotypic screen and let the cell biology tell us what the most important or most druggable targets are in that biology. And then finally, we're members of a large public-private consortium called ATOM, which is focused on using artificial intelligence and high-performance computing to do all of this much more rapidly. So I'll end with a little plug. The Small Molecule Discovery Center, as Shana mentioned, is a research collaboratory for chemical biology and drug discovery at UCSF. And we have 2 open positions. Encourage you, if you're interested in collaborative science and high throughput biology or high throughput biophysics, to talk to me about the opportunities in doing high throughput biology, high content imaging, cancer biology, complex in vitro models, and fragment-based drug discovery, particularly using biophysical methods in structural biology. And so with that, I'll just thank my lab. And I guess we'll go on to the next presentation. Okay, Lewis. Thank you. So it's a great pleasure today to introduce Lewis Kay. Lewis is a professor in the departments of molecular genetics, chemistry, and biochemistry at the University of Toronto. He is my estimation, a giant in the field of NMR for gigantic proteins. And his structure, function, dynamic studies of proteins involved in protein homeostasis have provided really very valuable and exciting insights into how these machines function. So with that, looking forward to hearing about how we can see the invisible NMR.
Thanks very much for the nice introduction and for allowing me the pleasure of speaking here today. After that introduction, I should say that I will not be speaking about gigantic proteins. These are going to be fairly small proteins today. And in particular, I want to talk about ways that one can go about, as the title says, seeing the invisible by NMR, and why that may be important in trying to provide insight into solving what I've called the drug problem. So I want to begin by considering a simple one-dimensional energy landscape that I show here. Obviously, this is simplified relative to what it would be in reality for a complex biomolecule, but it'll serve to emphasize the point. We as structural biologists tend to focus on this region here of the energy landscape, and for good reason. First of all, the majority of molecules that populate the landscape are domiciled in this region. And secondly, we have outstanding tools that allow us to get atomistic descriptions of these molecules that reside in this region here. But it may in fact be that function is dictated by structures of molecules that are of higher energy. For example, given that we have an energy landscape which is quite rugged, there's going to be molecules that are populated here and at higher energies, and these molecules may be important biologically as well. Unfortunately, one doesn't have to go very high up on the energy landscape, say a few kcals per mole, before one reaches a situation where the technologies that we have are no longer particularly good, because the populations of the molecules are incredibly small. So if you're a couple of kTs higher than the ground state, the population of the molecule, say this one here, which is resides here, might only be 1 or 2%. And then traditional methods are simply not terribly good. And so there has to be a change in paradigm whereby we begin to focus not only on ground state structures, which are obviously very important, but also on excited state structures that are going to be sparsely populated and transiently formed, because these may be the structures that are at least of some relevance in the drug discovery process. And the question is, how do we go about studying that which is invisible? And I never like to pass an opportunity to give a little bit of an NMR lesson. So that's what I'm going to do in the next few slides. How do we study things for which we can't observe them in spectra? So we have no direct data, if you like, for these invisible states. And I want to introduce that concept in terms of the following situation. Supposing that we're an enzymologist and we want to study how an enzyme might function, and the enzyme could exist in two flavors, a conformation red and conformation blue, and that those flavors exchange one with the other. And suppose that we cool down the enzyme to low temperatures so that there's no more exchange, so that we end up in an NMR spectrum, say, with a bunch of red peaks corresponding to the red conformation, and a bunch of blue peaks corresponding to the blue conformation. And then let's suppose that we have a 50-50 mixture, so we could see the reds and the blues. And rather than talk about individual peaks, what I'm going to talk about are these little spin buoys, which are moving at different frequencies depending on their color, corresponding to the different positions in the spectrum of these peaks. So this would be a simple situation to handle by virtue of the fact that we've got 50-50 populations, and so we can see both the red and the blue peaks. But what happens if we only had 0.1% of the blue stick buoys, so that we don't see the blue peaks anymore, and yet we want to get information about the blue conformation? So how do we go about doing that? And so let's first of all focus on our stick buoys that are not interconverting. So we've cooled them down such that, you know, runners are always runners and walkers are always walkers. And then what we do is we take all of these buoys at the start line and we allow them to evolve for a period in time, tau, during which the runners run and the walkers are going to walk. And then after the period tau, we're going to stop the runners and walkers and turn them around. Now, the runners have traversed a larger distance than the walkers, but if we give an equal amount of time tau that we gave in the first instance, then the runners are going to be able to catch up with the walkers exactly at a time 2 tau after we started this race. And we get what is called a spin echo after a Hun spin echo after a very famous physicist, Erwin Hun, at UC Berkeley. Now, this is a situation that exists if we have walkers and runners, and the runners are always running and the walkers are always walking. But of course, in reality, for molecules, we're going to have a stochastic interchange between one conformation and another. So in runner and walker speak, that means that the runners become walkers and vice versa stochastically. So how can we get out information about the system? Well, what we're going to do in a fixed interval of time, we're going to apply these pulses. Remember, a pulse is where we stop the runners and walkers and turn them around. And if we apply many pulses in a fixed interval of time at a rate that is fast compared to the differences in the speeds of the runners and walkers, then a runner will never get ahead of a walker. And that means that at the end of this interval in time, when we plot the distribution of runners and walkers that cross the finish line, we'll get a very narrow distribution. By contrast, if we only have a single pulse, so only a single period in which we stop the runners and walkers and then turn them around, we're going to get a very broad distribution. And if we apply 2 pulses or 3 or what have you, we're going to get distributions that vary in the broadness. So simply by measuring the fatness of NMR peaks as a function of the number of these pulses that we apply, we can get information about the relative numbers of walkers and runners, so thermodynamics of the system. We can get information about the kinetics, the rate of interconversion between runners and walkers. And we can also get information about the speeds of runners and walkers, or in NMR speak, we can get chemical shifts of, in this particular case, the invisible state that we're interested in. And the way that we show that in NMR experiments is to plot the fatness of our peaks as a function of the number of pulses that we apply. So that if we have essentially a flat line here, that would indicate that the probe that we're using doesn't sense an excited state. By contrast, if we get a line that looks like this, a profile that looks like this or like this, these are non-flat profiles, and they give us information about the kinetics, thermodynamics, and importantly, about the structure of the invisible state. So we get kinetics and thermodynamics of the interchange, but we also get structural information, which is very important. And so what one can do now is to construct a variety of different experiments, which we've done over the years, where we focus on essentially every atom in the protein, if you like, every nitrogen, every proton, every carbon. And we can, at different positions, measure these profiles, which gives us invisible chemical shifts, or the chemical shifts of the invisible state. And these chemical shifts are very valuable probes of structure. In addition, we can get other sorts of information like relative orientation of bond vectors, which gives us structural information. In fact, using a whole bunch of different experiments, we can devise ways, if you like, so that we can measure the same properties of our invisible state that you'd be able to measure of the ground state, for which you can see peaks in NMR spectra, but we just have to come up with different ways to do it because, of course, we can't see the peaks. There's another way that one can actually see the invisible as well, and this is a way that we stole from the MRI people and just modified what they were doing so that we could look at macromolecules and exchange within macromolecules. So suppose that we have a ground state and an excited state. We have a whole bunch of different probes that we can use, hydrogens, carbons, and nitrogens that are in the protein, if you like. And let's focus on a single carbon probe. Now, what we're going to do in order to study the exchange process is basically illustrated here. We're going to take a very weak magnetic field, which I call essentially a hammer, if you like, a green hammer, and we're basically going to tap all across the spectrum, looking for the invisible state. Now, I've shown the invisible state here in red, but in reality, you wouldn't be able to see it. I just show it here for educational purposes. So we have our little green hammer, and we tap. And then what we're going to do is we're going to look at the blue peak. That's the peak that is associated with the carbon probe in the ground state. Remember, we can see the ground state, so we focus our eye right on the blue peak, and we look for perturbations in the blue peak. So if we apply a tap far away from the ground state or the excited state peaks, nothing happens. Conversely, however, if we have our little magnetic hammer right on top of the excited state peak, our invisible excited state peak, then by virtue of the fact that there's exchange between the ground and excited states, that relays the information, the perturbation to the red peak, to the blue peak. And we see a decrease in the blue peak. And of course, if we take our hammer to the blue peak, we also decrease its intensity. And so what we're going to plot is the intensity of the blue peak, that's the peak that we can observe, as a function of the position of the hammer, the little green wave here. And we see a dip here that corresponds to the position of the excited state, which is now visible through the hammer. And then we have a big dip corresponding to the position of the ground state. And in this way, in fact, we can determine the positions of all of the nuclei in the excited state in a one-shot type of experiment and use that to get out structural information. And I just want to give you one example of the sort of structural information that we can obtain in the context of superoxide dismutase, or SOD1. Now, SOD1 is an enzyme that serves as an antioxidant, and in the mature form, it's a dimeric structure that's been elucidated by X-ray diffraction. And it's a very stable molecule. It's stable because it's a dimer. It's stable because it binds metals. It binds both a copper and zinc, one copper and one zinc per protomer. And it's stable by virtue of the fact that it has a disulfide bond, again, one per protomer in this dimeric structure. You can essentially boil this molecule without doing any harm to it. And nevertheless, this particular protein has been linked to ALS. Aggregates of SOD1 have been found in motor neurons, and misfolded conformers of SOD1 have been hypothesized to be the toxic species. And so we asked the following question: how can you take a super stable protein that you can basically boil and turn it into something that is responsible for disease, given its inherent stability? And so to begin to broach this topic, we decided that what we would work with is not the super stable form, the mature form of the enzyme, but rather the immature form of the enzyme, the form of the enzyme that lacks metals and lacks the disulfide bond and is only monomeric in solution. So this would be the form that is produced off of the ribosome initially before, of course, it's going to be modified and forms the stable dimer. And we asked the following question: how stable is this molecule in terms of order? One of the things that one can do with NMR spectroscopy is we can measure at each position in the molecule an order parameter, which is the amplitude of motions. An order parameter of 1 indicates that at that position, we have a very ordered structure. Conversely, 0 indicates that the structure is very much disordered. In red, what I show here, the order parameters is a function of amino acid along the polypeptide backbone. We have order parameters that are roughly 0.9. So a very stable structure for this dimeric conformation. Conversely, if we look at the baby form of the enzyme, in green, you can see that the order is much less. And in fact, in regions that are loopy, such as this zinc loop and the electrostatic loop, you can see that there's a lot more disorder. Now, the beta strands are still formed in the nascent structure. We know that because we can predict interactions from the dimeric structure shown here in blue, predicted NOEs, predicted distances, if you like, and then the observed distances in the baby structure are shown here in red. You can see that there's a very nice correlation. So the beta strands are still present, but the loop regions are much more dynamic. And so what we want to do is we want to study conformational fluctuations from the ground state in this baby structure. And when we do that, we discover that there's a very rich energy landscape. So our ground state, our populated state is going to be shown here. This is the structure. And now what we're going to do is we're going to use the stick boy experiment and the hammer experiment to interrogate the energy surface, the energy landscape, and to see what the transitions from this ground state might be to excited states. What are those excited states structurally? That may provide us with some insight into how, for example, this ground state may give rise to aggregates. And so what we've done, and this is shown here an example from the hammer experiment, we've noticed a number of different processes, a number of different excited states. So if we focus on the hammer experiment, we have a process which we call process 1, which is revealed by, for example, glycine 61 and the backbone amide of glycine 61, although there are many residues which give us insight into this process. Well, you can see here that we have this dip here. This is the minor state dip, which indicates that there is an excited state. You can see that as a function of concentration in red, that's a concentration of 1.3 millimolar, in green 0.7 millimolar. So as we increase the concentration, you can see that the excited state is also going to increase. So we know that the excited state corresponds to some sort of aggregate. It's more than a monomer. We start out with a monomer in the ground state, but we get something bigger. And then what we've done is we've taken all of the nitrogen, all of the carbon chemical shifts of the excited state, and we've asked the question, what do these chemical shifts tell us about what the excited state looks like? And what I show here is a plot of the chemical shifts of the excited states that we measure via the hammer experiment versus the chemical shifts of the same nuclei in the very stable dimeric form of the enzyme. And you can see that there's excellent agreement between these chemical shifts, indicating that this process, process number 1, is one that takes this baby molecule and essentially generates a stable dimeric structure. So this is one way, if you like, that the baby molecule grows up. We also notice a second process, which we term process 2. Again, we visualize it via the hammer experiment. It's no longer a concentration dependent, so it's a process that happens within the monomeric state. Again, if we measure nitrogen and carbon chemical shifts that are associated with this process, and we plot these chemical shifts as a function of the shifts in the stable holoform of the molecule, we again see a very nice correlation. And this allows us to link the conformational change with the formation of the helix shown here. This is a helix that is part of the electrostatic loop, and it's an important helix in conferring the binding properties, the zinc metal binding properties to the structure. And in summary, one can not only notice native-like conformation, so this molecule progressing to form a native sort of structure, but in addition, we notice a number of aberrant conformations, which are dimeric in nature, and for which we can design models that may indicate that these are the initial springboard points, if you like, for formation of high molecular weight aggregates. So all of this work was done starting from the baby nascent structure, the wild type form. But what we decided to do is we would look at mutants of this superoxide dismutase and see how that might affect the structure of the nascent form as well. And so what one can do in NMR is simply measure chemical shifts of the wild type and compare those with disease mutants. If the chemical shift changes are very small, then there's no change in structure. And that's what we noticed, that for a half a dozen mutants or so, the chemical shift changes plotted along the y-axis here as a function of position are really very small. We see small blips. This is a large blip, which is at the location of the mutation. So if you make a mutation in the vicinity of the mutation, you will get chemical shift changes. But there are really very small chemical shift changes throughout the molecule, indicating that the ground state structure doesn't change when you invoke disease mutations. But if you go up a little bit higher in energy, there are significant changes that accompany disease mutations with the excited state structure. So that, for example, if we look at process number 1, where we have superoxide dismutase, again, we're focused on the nascent polypeptide chain, and I indicated that it forms a dimeric structure roughly 3% of the time with a lifetime of a few milliseconds, as indicated here by this dip. This dip disappears completely in a number of disease mutants, indicating that this dimer is no longer formed. And additional experiments that one can do indicate that other processes are also going to be eliminated in some of the disease mutants, but not in others. In addition, various processes are actually formed in disease mutants that weren't there in the wild type. For example, if we look at the G85R disease mutant, in red, I show now the stick boy experiments. You can see that these profiles are not flat. That indicates that there is a conformational exchange process occurring. Conversely, in blue, I show the results for the wild type structure, indicating that there is no exchange occurring for the wild type. So the conformational landscape can become both simpler and more complicated with these disease mutants. And we can localize what the changes in structure are on the basis of these sorts of experiments. In this particular case, for both G85R and G93A, the excited state is one that is affects a couple of strands, beta 5 and beta 6, which tend to open up. And this might be important, again, for aggregation. So let me just conclude by saying that the story that I've told you about today, involving the use of stick boy experiments and hammer experiments, is not only applicable to superoxide dismutase. Other laboratories have looked at other proteins that are involved in aggregation and have shown that you have small molecules, or sorry, small populations of conformations that are formed that give rise to fibrils. Also, in the context of DNA, spontaneous mutations can be incorporated because of excited states that are formed, and these excited states are not detected via proofreading mechanisms that we have. Perhaps a stunning example was generated this year in Science on Abl kinase. Abl kinase, obviously an important molecule, an important drug target against leukemias. Not only is there a ground state, which is 90% populated, there are a pair of excited states that were determined by the hammer experiment. These excited states are inactive insofar as the kinase is concerned, and they're primed for binding various drugs that we use in the treatment of CML. Various cancer mutations, however, which abrogate the function of the drugs, have been shown to decrease the populations of the inactive states and therefore prevent binding in this way. So I think that there's a real opportunity not only to focus on the ground states of these molecules, obviously that's important and it's fairly easy to do, but also to reach sort of beyond the ground to the sky, if you like, and to focus on excited states of molecules as an opportunity really to understand how it is that we might drug these molecules effectively, and also when there are mutations which prevent druggability, what's going on. And let me just conclude by acknowledging the people who have done the work. Ashok was involved in the SOD1 project, which was a collaboration with Liz and a couple of former postdocs in the lab who were very important in the development of the methodology. So I'll stop there.
Oh, fantastic. Thank you. Thanks so much, Louis. Okay, I guess if you stop sharing, and now we have Jason Gestwicki. So Jason, well, oh, great. So it's a pleasure also to introduce my colleague, Jason Gestwicki. Jason's a professor of pharmaceutical chemistry. He's also the director of the Chemistry and Chemical Biology graduate program and the associate director for academic affairs at the Institute for Neurodegenerative Diseases at UCSF. His research is focused on chemical biology and drug discovery for protein chaperone networks, particularly HSP70. And his chemical probes and drug leads have given us a lot of insight into diseases and how to treat certain diseases like cancer, Alzheimer's disease, even cataracts. He started several companies around this, including Viewpoint, which was focused on cataracts. And so with that, Jason, please take it away.
So thanks so much, Michelle, and thanks to you and to Shana for including me in this. It's an honor and a lot of fun. And so I want to tell you a short story from the lab today. Maybe before I get started, we'll just start with a little overview on my own view, or our view on protein homeostasis in my lab. And so just to kick it off, the proteins are born. They're born off the ribosome. They're born in this sort of noodly type conformation, which would have been the high energy conformation on Lewis's diagram. And then much of them need to fold into something like the native state or those high energy states that Lewis was showing. And so that process in the cytosol is managed, and it's managed by a series of about 180 molecular chaperones. And those chaperones recognize those folding intermediates. They help proteins fold. They understand when that protein has misfolded through molecular recognition. They give the chance for proteins to disaggregate. They give chance for a protein to fold and unfold. For instance, if they have to traffic over or translocate through a membrane, for instance, they might need to unfold and then refold on the other side. And so the chaperones manage the balance of protein synthesis, folding, quality control, and then turnover. They couple to the autophagic system, and they couple to the ubiquitin proteasome system, and manage how proteins are made, folded, trafficked through the cell, assembled into complexes. And so we got really interested in this because in some ways, 180 molecular chaperones in the human genome in some ways seems like a very small number to be able to manage that process for the entire human proteome all the time. And so we got really interested in trying to use our interest in chemical biology to figure out how these things work. And so here's a quick diagram, a cartoon diagram, overview diagram of the molecular chaperone network with a subset of these 180 molecular chaperones. And a lot of you, especially from the University of Toronto side, will know of Lewis's great work on the heat shark protein 70 system. I didn't check in the participants list, but you'll probably also know if you're in Leah Cohen's group on the Toronto side or Miko's group on the Toronto side, you know a lot about heat shark protein 90, and you've done a lot of great work on the heat shark protein 90 interactions. There's a whole zoo of these molecular chaperones, small heat shock proteins, these large complexes, these DNA J proteins. And one of the key features, I think, the feature that drew me to this system are these red lines between these cartoons. So these red lines between the cartoons are direct physical contacts between molecular chaperones. This is where molecular chaperones bind directly to each other. So heat shark protein 70 might bind directly to heat shark protein 110, for instance. I don't think that's an accident. And the basis sort of our program is that that's probably not an accident, that these 180 chaperones, in order to do their job of managing protein homeostasis or proteostasis, they need to work together as large complexes with direct physical contacts between them. And so maybe by an analogy, hopefully this will help some of you who don't think about this a lot, an analogy of how we think about how the logic of the system is set up is to take this sort of diagram with these molecular chaperones and think about it like this. Think about it like a subway system in a major city. This happens to be London, but it could be anywhere. The infrastructure of a subway system is the physical interactions between molecular chaperones. That forms the tracks on which the people are trafficked from one side of the city to the other. They want to be able to interact with a molecular chaperone or a specific subway at one location, know how many stops they have to go through, get handed off from chaperone to chaperone, and then end up being translocated into endoplasmic reticulum or degraded through the ubiquitin proteasome system. And so in some ways, the clients or passengers in the cell are trafficked through this web or network of molecular chaperone protein-protein interactions. And the basis of my lab, I think in the spirit of how we're trying to set this seminar program up today, the spirit of what my lab tries to do is take this molecular chaperone network, these direct physical interactions between chaperones, structurally and biophysically characterize these different red lines, the connections between chaperones, and then develop chemical probes that either break those contacts or promote those contacts. And then use that chemical probe after a long series of medicinal chemistry, like Michelle Arkin introduced, use those chemical probes to come back in, break connections between specific stops on that subway system, and then understand what happens. Where do people go if they can't be trafficked to the ER anymore, or can't be trafficked to the UPS system? What's the logic of this? Can we use chemical probes to break or form different connections between chaperones and thereby work out the logic? And as Michelle said, this process is also disrupted in many different diseases, cancer and neurodegeneration and other diseases. And so at the same time, we're trying to understand the logic by chemical biology. Sometimes, eventually, we get lucky and we find a molecule that might perturb the system in such a way that it actually might be a treatment for a specific disease. And then we can pursue the translational opportunities. And so in the spirit of this program, I'll go through one specific example here that I thought you might be interested in, a recent example from the lab. But hopefully, everyone can think about their own specific interests here, and maybe we can build some collaborations, as Michelle indicated, between Toronto and UCSF around targets that you're interested in. Okay. One of the reasons, I didn't do my training in molecular chaperones, but one of the reasons I got interested in doing it when I started my own lab about 15 years ago or so is because molecular chaperones are a really, really rich source of really diverse protein-protein interactions. And so here's a couple of snapshots of this zoo of molecular chaperone protein-protein contacts. One of the molecular chaperones will be in green. The other chaperone will be in gray in each case. And then I've plotted out some of these on the affinity of that protein-protein interaction and the buried surface area in that protein-protein interaction, and put these quadrants on just to guide the eye. And you can see, even from just a snapshot of the pictures, these protein-protein interactions are all over the place. Some of them take up a very large surface area, some of them are very small, some of them are very tight, some of them very weak. And importantly, for what I want to tell you today, some of them involve helices interacting at surfaces, some of them involve beta sheets interacting at surfaces, some of them involve loops interacting at surfaces. And so this one set of interactions in the cell, this one set of protein-protein interactions in the cell gives us as chemical biologists a really rich set of targets with really diverse topologies that we can use to build techniques, technologies, high throughput screening approaches, and different ways of breaking protein-protein interactions, even within this one type of biology. And as I mentioned, most of the interactions in this system, like in most systems, are mediated by turns or sheets or helices. But that's not what I want to talk about today. I want to talk about a new thing that we're really interested in the lab. And that's the specific type of interaction that occurs at termini. So at the N termini of a polypeptide or the C termini of a polypeptide. This is a neat part of the protein to do a protein-protein interaction because the chemistry is really unique. The pKa's and the chemistry of the amine terminus and the carboxylate are really unique. And then the topology is, of course, really unique because you have a long sort of usually disordered noodly type N-terminal or C-terminal part interacting usually with a folded domain. And so this is a neat place to do inhibitor design for protein-protein interactions. And so we have a number of projects going on in the lab on this type of protein-protein interaction. I'll tell you the story of Matt Ravlin from my group, who recently published his work on the interaction of heat shock protein 70 and 90 with the TPR domain co-chaperones, which I'll get into next. But I want to quickly mention it again in the spirit of this symposium. And we follow Lewis's work in the 20S proteasome. He's a leader in that field very carefully. We've gotten really interested in the interaction of the 20S proteasome with activators. I wanted to mention that work too. And then we've also got really interested in this interaction of these JDP proteins with HSP70s. And I know, for instance, Miko is interested in this area as well. So I thought I would mention that for broad interest. But I'll just, in the interest of time, I'll just focus today on Matt Ravlin's work. And that work involves specific protein-protein interactions here between these little tiny ends of heat shock protein 70 and heat shock protein 90 with these TPR domain proteins. We've been working on these for a long time. HSP90s and HSP70s in the cytosol end in these amino acids EEVD, where the HSP90s have MEEVD and the HSP70s have IEVD, followed by the true carboxylate. So we've got this really interesting chemical environment here where there's a carboxylate from the aspartate here and then the true carboxylate of the polypeptide. And that interacts with these TPR proteins through what's called the carboxylate clamp, where you have these cationic side chains, lysines, arginine residues, etc., that form the series of hydrogen bonds that recognize this unique orientation of those two carboxylates. And in the interest of time, I'll just really briefly mention this, guided by David Toft and many others and Lynn Regan, we and others have found that the EEVD motif has almost 80% of the binding energy of this protein-protein interaction between HSP70s and TPR domain proteins. So most of the interaction energy actually comes from that aspartate. Interestingly, we and others, Lynn Regan has done the classic work here from Yale, this M versus I in the HSP90s and the HSP70s turns out to be really important for generating selectivity. And here's the data, but I'll just summarize it. I'm happy to talk about it in Q&A if you're interested in. But the HSP70s are here, the HSP90s are here, and both series bind really well to a subset of these TPR domain chaperones, but only the HSP90s really bind to these FKBP 51 and 52s. So this got us interested in this a few years ago because there's some generality in this protein-protein interaction, but then some selectivity too, which is kind of cool. Even from this one residue difference between the ends of these carboxylates, you can generate some types of selectivity in this system. And so from that type of protein-protein interaction mapping and all the different KDs that we measured, you could build a little subnetwork, this little part of the subway system, just focused on how HSP90 binds all these different TPR proteins and how HSP70 binds a subset of those TPR proteins. I'm going to talk specifically today about Matt Ravlin's work on an E3 ubiquitin ligase called CHIP, which interacts with both of them. And CHIP is really important because it'll couple to both HSP90 and HSP70. And when it does that, it will polyubiquitinate client proteins that are bound to those chaperones, ubiquitinate those and take those for degradation. But if you instead of having CHIP, if you have FKBP51 bound to HSP90, for instance, then you'll do proline isomerization. FKBP is a proline isomerization enzyme. PP5 is a phosphatase. So if you have PP5 plus HSP70, that will dephosphorylate substrates. And so this protein-protein interactions is a particularly interesting part of this molecular chaperone network that really drives what's going to happen to that client or that passenger in the network. Okay, so the first question we wanted to ask, and we did with Charlie Craik at UCSF, was we really wanted to understand, build off of this observation that the M and the I residue in HSP90 and HSP70 seem to have selectivity for a subset of chaperones. That got us thinking, does a specific TPR protein like CHIP, does it have a selective set of ligands? Can we generate selective inhibitors of just CHIP within this entire network? Harkening back to Michelle's talk on P97, could we come up with a molecule that just breaks one specific strategic part of that larger network and leave the rest of it untouched? And so we used this technology from Charlie Craig's group, in which we generated pools of peptides by automated robotics peptide synthesis, such that we can sample about 640,000 peptide sequences, keeping that D that's really important at the end for the dicarboxylate clamp. This is published, so I'll go over it quickly, but we measured the interaction affinity of every single peptide in these pools for CHIP's TPR domain, and generated a logo plot and found that indeed we could generate a much tighter binder to CHIP's TPR domain, much tighter than the native sequences. So here's just a fluorescence polarization experiment in which we measure binding of the native HSP70 and HSP90 sequences. Those bind with micromolar affinities or so, but then we could get 2 orders of magnitude tighter affinity for our optimized sequence by selecting the best amino acids from these pools. And then we did alanine scans, figured out where the interaction affinity comes from, and solved 2 crystal structures, one with the native sequence and one with the optimized sequence, which we call ChIP-opt, because it's optimized specifically for CHIP's TPR domain. In the interest of time, again, I won't tell you all of the details, but we're really excited to see things like this tryptophan residue binds a nice hydrophobic pocket here, giving tighter binding than the native sequence. And really excitingly, this proline in the second position here, the P2 position, just upstream of the aspartate, that generates a really nice pre-orientation of the amino acid backbone here to, again, give you much tighter binding to CHIP's TPR domain. And then there's a few other new contacts that we picked up in this ChIP-opt versus the native HSP70 sequence. Matt Callahan, a graduate student in some unpublished work, has asked the question of whether ChIP-opt is also selective for CHIP's TPR domain in cells, not just by these affinity measurements by fluorescence polarization. And he built a system in which he built a probe with a photoactivatable group and then a biotin, so that we can pull down everything that binds these I or M EEVD sequences from cells or cell lysates. And in bold here, this is the mass spec data from his SILAC experiments. And in bold here, indeed, he was able to pull out almost all of the different TPR domains from these cells. Using that type of assay, then he could add the ChIP-opt peptide and see which of these interactions would be blocked by the ChIP-opt peptide. And this is just super cool, I thought. And Matt did a great job on this. And we were both really excited. Indeed, this ChIP-opt peptide now is really selective for blocking the interaction of CHIP's TPR domain, but it leaves alone all those other TPR domains. So now we can selectively interfere with that part of the network, but leave all the rest of the network really untouched. So we're still working on building a really good chemical probe and doing the medicinal chemistry, and a new postdoc in the lab, Jeanette Bertrand, is leading this up, and I'm really excited about her work. But just as proof of concept, a postdoc in the lab, Corey, built a luciferase complementation experiment where we could look at the TPR binding to this EEVD motif in cells by luminescence, and then he made a peptide that has a cell-penetrating peptide attached to it, and indeed that will block the protein-protein interaction in cells. And so based on all of this, we're building chemical probes at the moment to ask questions about when and where this HSP70, HSP90 CHIP interaction is important. I wanted to leave you with a few things. I'm going to do a really quick job of this, and I apologize, but I just want to plant some seeds, again, in the spirit of this sort of introductory, collaborative Toronto UCSF combination. There's other things we can do with this information, and we can flip this question and ask, using all of this biophysical information, we can ask, what else does CHIP bind to in the cell? And so we had all of this affinity constant information from fluorescence polarization and differential scanning fluorimetry. We had all this information about which amino acid preferences CHIP's TPR domain had. And so we can use that to build a scoring function that basically takes any 5 amino acids and predicts what the affinity for CHIP would be, which we call a CHIP score. And then we can compare that to gold standard, actually measured, experimentally measured affinity constants, and find that we get pretty good prediction. So if you send me, you can send me an email and you can say, hey, I have these 5 amino acids. Do they bind to CHIP? We can give those a CHIP score. And then we can use that to blast off the human genome and say, what other things in the human genome at the C terminus, what other 5 amino acids at the C terminus bind really well to CHIP? And we could do that, and we can find other things that bind better than HSP90 in the human genome. And we find a couple. And if there's anybody from Miko's lab on the call, I really hope there is. These two right here, these arrows, actually Miko found binding to CHIP by mass spec, or by his, I'm sorry, by his Luminaire technology. And so we really totally independently came up with those protein-protein interactions that Miko had found. But there's a whole bunch of other ones in here. And this is my major point, is that we're launching a lot of cool projects now, not by necessarily blasting for the EEVD sequence in the human genome, but using these biophysically based scoring functions to blast the human genome to find potential new protein-protein interactions with this TPR domain chip or any other type of interaction that we're interested in. Okay, and I'm going to leave this part. This is published. This is a great collaboration with Michelle Arkin's group in which we mapped some of these interactions, but I want to leave time, plenty of time at the very end for broader discussions here. And so I'll just leave you with a couple of take-home messages that I think are important. We're really interested in protein-protein interactions. We're interested in protein-protein interactions because they're the backbone of the molecular chaperone network. They're the things that stitch together the molecular chaperone network and allow proteins to interact with that network, be handed off from one chaperone to another chaperone to another chaperone to control protein homeostasis. We think there's a lot of really interesting drug targets in this network that we could use to change how proteins are folded and how they're degraded in the cell, both for neurodegeneration and for cancer and other types of diseases. We like these PSSCL peptide libraries for mapping absolute selectivity of these protein-protein interactions. And we're really interested in these N and C terminal interactions, or interactions that occur at peptide termini right now. I skipped over a lot of this part, but we think we've learned a lot of cool things. We've launched a lot of cool projects in the lab around CHIPS interactions using this technology. And then more broadly, hopefully for the entire group, we think this structural and biochemical knowledge can be used to build new protein-protein interaction inhibitors. And so I tried to point these people out as I went, but just in case, Matt Ravlin really led this project jointly between Charlie Craig's group and Michelle Arkin's group and a pathologist who I didn't mention, Leah Grinberg here at UCSF. And then Corey, a postdoc in the lab, and Matt, a graduate student, have taken this project into really exciting new directions. And Jeanette is taking this information to build inhibitors of the specific protein-protein interaction. So again, I want to thank Shana and Michelle for having me. It's great to have a chance to talk to you today. And I'm happy to answer questions at the very end of these seminars, or if you want to drop me an email, please do, and I'm happy to talk to you about any of these targets. Thank you very, very much.
Thank you, Jason. Fantastic. It's great to, I would describe that I know this group of people pretty well, and yet I have no idea what they're going to talk about. It's really exciting. And so I'm sure John will be no exception. So John Rubinstein is a native of Toronto. He started his own research group at the Hospital for Sick Children in 2006. He holds a Canadian research chair there and studies the structural biology of bioenergetics, develops new methods in CryoEM to facilitate this work. He's published several beautiful and insightful structures on molecular machines in the AAA family. And though I'm not really sure what he's going to talk about, I'm sure it's going to be equally insightful and stimulating. So John, take it away. Are you muted, John?
There we go. Sorry. Sorry about that.
It's my fear that I've given a whole talk muted. Nobody's told me so.
Well, that problem's taken care of. I'm delighted to be here. I was actually thinking about something I learned on the weekend as Shana was introducing the Connaught Global Challenges program, and that is that in the 1950s, the mass production of polio virus that enabled the vaccination of millions of people with a polio vaccine was actually led by the Connaught Laboratory in work done in the group of pioneer woman in science named Leonie Farrell. And we're really hoping that that sort of effort can be parallel to end the current pandemic. In my own group, we've spent the past 15 years since I've established the group not only developing new methods for CryoEM, but also establishing our own pipeline for solving structures of proteins in a relatively quick time frame. So at the moment, the way that pipeline looks like is something like this. It's overseen by a former postdoc at the lab, Samir Benlikber, who's now a facility manager for a large chunk of the CryoEM infrastructure in Toronto. And what we have is really good access in the lab to a low-end microscope that we can use for screening specimens and optimizing cryo-EM conditions. After we've optimized conditions, we take our optimized grids to a shared resource. So this is shared in that really large Toronto community between the Hospital for Sick Children, the University Health Network, and the University of Toronto Faculty of Medicine. And in a short time, we can collect a high-resolution data set of those specimens that we'd previously optimized. And due to computational advances, we can produce high-resolution maps of protein complexes, really just taking weeks from the time we obtain the purified protein sample to the time we calculate a high-resolution map that we can then use to build an atomic model. The biological theme of the laboratory is the membrane protein complexes that exist at energized membranes. The idea of a membrane that can maintain a transmembrane proton motive force was put forward in the 1970s and 80s by Peter Mitchell. Peter Mitchell was really the patron saint of an activity that many of us take part in right now, which is working from home. Unlike many of us, Peter Mitchell was actually extremely rich, and the Glynn Laboratories, that is listed as his affiliation for his Nobel Prize in 1978, was actually his own house in Cornwall, England, where he carried out all of his own experiments. What Mitchell proposed was that in biology, energy can be stored as a transmembrane proton motive force, which was really a paradigm change in biology, because up until that point, people really thought of cells as bags of enzymes. But with Peter Mitchell's chemiosmotic hypothesis, the cell suddenly became divided into compartments that can maintain a transmembrane proton motive force, or really a transmembrane ion gradient. And that really defines our view of how we think of cells nowadays. The biological context that Mitchell was working in was that of oxidative phosphorylation, the production of ATP in aerobic cells. He was thinking about the electron transport chain, where NADH, produced by the Krebs cycle and fatty acid oxidation, is used to transfer electrons to complex 1, reducing a pool of quinone to quinol, and that quinol pool then donates electrons to complex 3, and then complex 4, ultimately reducing oxygen to water, with a reduced quinol pool also contributed from the oxidation of succinate to fumarate. That transmission of electrons through the electron transport chain is also coupled to the pumping of protons from one side of the lipid bilayer to the other side of the lipid bilayer, setting up that proton motive force that Mitchell proposed. The ATP synthase dissipates the proton motive force and uses the free energy liberated from the dissipation of that proton motive force to power the production of ATP from ADP and inorganic phosphate. Another way that cells can produce a proton motive force is with the vacuolar type ATPase. It looks a lot like the ATP synthase, but it operates in the opposite direction. It hydrolyzes ATP to ADP and phosphate and uses that energy to pump protons. I'm not going to talk about the vacuolar ATPase in my talk today, but I do want to acknowledge a great deal of work by really about half the lab over the past 7 or 8 years, where we have solved a number of structures of vacuolar type ATPases, work done by Jinhua Zhao around 2015. Jinhua is now a postdoc at UCSF and on the job market. Mohammed Mazhab Jafari, who got our first high resolution structure in 2016. He's now faculty at UHN. And then current PhD student and postdoctoral fellow, Thamia Vasanthakumar and Yazan Abbas, who also determined high resolution structures of the yeast VATPase and mammalian VATPase. And we've learned quite a lot from these structures that do inform on the subject of my talk today, which is the ATP synthase. For about the past 5 years in the lab, all of our work on the ATP synthase has really been spearheaded by Hui Guo, a current PhD student. And Hui has been able to determine quite a large number of structures during his PhD, initially starting with the first structure of the membrane region of the mitochondrial ATP synthase. And because this was the first structure of the membrane region, and we already knew the high resolution structures of the catalytic region, Hui was able to build this first atomic model of the intact ATP synthase. He subsequently moved on to solve the first structure of a bacterial ATP synthase, and because he was now using newer computational methods that let him solve the structure in the intact enzyme while focusing on one region or another computationally, he not only solved the structure, but solved the structure in multiple conformational states. So in the activity of an ATP synthase, protons will go through the membrane-bound region, and the passage of protons through that region will actually induce conformational changes in the membrane-bound region that drive the rotation of this rotor complex that lead to the synthesis of ATP from ADP and phosphate in this catalytic region. Now, Hui was working on the canonical bacterial respiratory pathway, and prior to that, the canonical mitochondrial respiratory pathway. However, in the lab, we have worked on a wide variety of organisms, many different bacteria, many different fungi, and a variety of mammals. And once Hui was able to get this first structure of the canonical bacterial ATP synthase, we realized it was time to turn to another type of organism, and that is mycobacteria. This genus has a very different electron transport chain system. And as you know, mycobacteria are a very important genus of human pathogens. So because of these differences, we realized that we ought to start looking at the respiratory system of mycobacteria as a drug target. Now, this was actually a justified approach. In 2015, in 2012, a first new drug for tuberculosis had been approved by the FDA. It's the first new drug for tuberculosis in more than 40 years. And it turns out that that drug targeted the respiratory pathway in mycobacteria. So this was a validated pathway for anti-tuberculosis drugs. And tuberculosis, as you will know, is a very important human disease, the leading cause of death by infectious disease in people, responsible for about 1.5 million deaths a year, with over 200,000 childhood deaths and increasing antibiotic resistance. So there's an apparent need for work on new drug targets for anti-tuberculosis drugs. And our initial activities for looking at the mycobacterial respiratory pathway were really due to a collaboration that arose with a colleague and good friend Peter Brzezinski in Stockholm University, where Peter's lab was able to isolate the mycobacterial respiratory supercomplex. This is a large assembly that functions equivalently to complexes 3 and 4 in mammals, but forms an obligate supercomplex with a bound superoxide dismutase domain in mycobacteria. And so now that we knew that mycobacteria were important and we had started doing some experiments with mycobacteria, we realized it was time to actually look at the true target of this first drug that had been identified as a new antimycobacterial drug. And that drug is a compound called bedaquiline that targets the mycobacterial ATP synthase. Bedaquiline is a very important drug. The organization MSF estimates that 400,000 people should be taking bedaquiline each year. And now that the pharmaceutical company Janssen has decreased the price of bedaquiline dramatically, that drug actually will become accessible to, if not all 400,000 people, but a larger fraction of those people. So our first attempt to get the mycobacterial ATP synthase was due to the effort of my lab manager, Stephanie Buehler. And Stephanie tried the approach that had worked so well for Hui in getting the canonical bacterial ATP synthase. She took a plasmid that had the entire codon for the ATP synthase on it, inserted it into E. coli that had the endogenous ATP synthase knocked out, and tried to express the protein and purify it. And that worked to some degree. This is a negative stain image of some ATP synthase particles, but this is actually not a very good negative stain image. This average of particles actually hides a large aggregate, and it is very hard to produce a lot of protein, and much of the protein was aggregated. But what it was sufficient for was writing a grant application where I could insert this image, and the grant application was funded, and we got Stephanie a cake for her effort, and that gave us the money to pursue other methods of getting the mycobacterial ATP synthase for structural studies. It turns out the method that worked in the end was due to help we received from Jun Liu, who's a mycobacterial expert in the Department of Molecular Genetics at the University of Toronto. And I won't go into the details of the procedure, but it essentially gives you a toolbox for inserting things into the chromosomal DNA of mycobacteria. So Stephanie made a first strain where we were able to purify the endogenous mycobacterial ATP synthase by growing up large vats of Mycobacterium smegmatis, a non-pathogenic, fast-growing relative of Mycobacterium tuberculosis. And Hui, again, with a new, starting with a summer student, but then a PhD student who's about to finish his first year, Hui and Gauthier Courbon were able to obtain these beautiful cryo-EM images of the mycobacterial ATP synthase. Actually, at this point, using a strain that Gauthier had made that had been further optimized for the purification of this protein. And at a first glance, the mycobacterial ATP synthase really looks quite similar to the canonical ATP synthase. So it has many of the same subunits. There are a few differences. For example, this peripheral stalk structure in most bacteria consists of 2 different subunits. In mycobacteria, sorry, it consists of 3 subunits. In mycobacteria, it consists of 2, where 2 of the canonical subunits have been fused to form what we call this B delta fusion protein. It's even more similar when you look at the subunits in the membrane, where proton translocation is coupled to rotation of this rotor subcomplex. For those subunits, we could really overlay it with the canonical model from Hui's structure of the Bacillus PS3 ATP synthase, or a subsequent structure of the E. coli ATP synthase that came out. It's basically an indistinguishable fold. However, there's a very important property of ATP synthases, and that is they need to be able to prevent the reverse activity. If a cell loses its proton motive force across the membrane where the ATP synthase resides, the ATP synthase will actually run backwards and hydrolyze ATP and depletes the cell's supply of ATP, leading to the rapid death of the cell. In mammals, there are actually extra subunits that will bind and prevent ATP hydrolysis during ischemia to prevent ischemia reperfusion injuries. But in bacteria, there are no additional subunits, and it's the ATP synthase itself that must somehow prevent ATP hydrolysis. Hui's experiments with the Bacillus PS3 ATP synthase showed us how it happened in that enzyme. Hui found that this subunit over here, the epsilon subunit, actually has an alpha helix that extends up in between the catalytic subunits. And if I show you a cross section of these catalytic subunits, you can see that the epsilon is positioned so that if the enzyme were to turn in the ATP synthesis direction, there's no problem. There's nothing blocking this epsilon subunit. But if there's not enough of a proton motive force to support ATP synthesis and ATP hydrolysis begins to occur, the epsilon subunit interlocking between these catalytic subunits will actually bump into one of these catalytic subunits and prevent the rotation in the ATP hydrolysis direction. So that explains how ATP hydrolysis is prevented in the canonical ATP synthase, but the mycobacterial ATP synthase appeared quite different. The alpha helix of that epsilon subunit is not in that upward-facing conformation that prevents ATP hydrolysis. However, when we purified the mycobacterial ATP synthase, similar to the other ATP synthases, it didn't show ATP hydrolysis activity. Instead, what Hui and Gauthier noticed was an extension of one of the catalytic subunits that came down out of its normal position and actually ran alongside this rotor part of the enzyme in each of the main rotational states that we observed. So this extension of this red subunit actually serves as a cross-link between the static part of the enzyme and the rotating part of the enzyme. And when you look in detail at that interaction, you can see that that red extension actually goes underneath some of the alpha helices or some of the sequence of this rotor subunit to act as a hook, sort of like the hook of a ratchet. So based on that, Hui could try an experiment where we inserted the affinity tag that we used to purify the enzyme. Really, the chromosomal modification technique at the time was the only genetic tool we had. So if you only have one tool and that's a hammer, all problems look like nails. So we used our hammer on this nail, and we inserted the affinity tag in a position in the chromosomal DNA of the smegmatis so that it truncated this alpha extension. And when we purified the complex with the truncated extension, we saw that it had really robust ATP hydrolysis activity. So our idea for how the ATP synthase prevents ATP hydrolysis is this. With this extension bound to the central rotor, you can see it's quite taut, and you can't turn the rotor in the hydrolysis direction, but you can turn it in the synthesis direction. In fact, Hui and Gauthier noticed an additional conformation of the enzyme where in one of the rotational states, that extension was sort of doubled back on itself And it looked like it's in a position where you could turn one click in the hydrolysis direction. And if you were to turn in the synthesis direction, you would disengage that hook from where it's attached to the central rotor. So that leads to a model that looks like this, where ATP hydrolysis would cause that extension to bind the central rotor. The enzyme could turn one click in the hydrolysis direction, but it can't turn any further. When the protomotive force is reestablished, the enzyme begins to turn in the synthesis direction. That leads to the folding over of that extension, and then the extension can be released so the enzyme can continue to run in the synthesis direction and make ATP until the protomotive force collapses, and then that extension would rebind. And of course, mycobacteria, such as Mycobacterium tuberculosis, often have to survive low energy conditions in the nodules that form the lungs and can persist as latent disease for decades. And this efficient inhibition of ATP hydrolysis provides a mechanism for them to prevent or conserve their energy. So that led us to this drug, bedaquiline, which is this important therapeutic used currently for the treatment of multi-drug resistant and extensively drug-resistant TB. And it was known from mutation studies that bedaquiline binds one of the subunits in the membrane region of the complex. The drug inhibits ATP synthesis with a nanomolar IC50, and if you make a mutation in one of those membrane-bound subunits, you can prevent the inhibition of ATP synthesis by the drug. Now that we had a way of actually inducing ATP hydrolysis by the enzyme, we found that the drug also had a nanomolar IC50 for ATP hydrolysis. And that was interesting because it's actually in conflict with some earlier studies where the affinity of the drug and the target of the drug, or what was perceived to be the target of the drug, that ring-forming subunit in the membrane, was measured, and it was only in the micromolar. And a crystal structure was determined with millimolar drug and these rings of subunits, and that really led to a disconnect between the nanomolar activity in vivo and ATP synthesis and ATP hydrolysis, but the micromolar affinity of the drug and its known target. So we thought that looking at the structure of the drug in the intact ATP synthase could show us that it had different drug binding sites. And indeed, you could see there are drug binding sites that just involve this ring, but there are two drug binding sites that also interact with this green subunit that induce rotation of the ring. So the drug binding sites that were seen previously only with the ring appear to be quite shallow, but the sites that involve the green subunit that was not seen in the earlier structures are much deeper. And we reasoned that these deeper sites would provide closer contacts, and those could be the high affinity nanomolar sites that are actually used to prevent ATP synthesis or hydrolysis in vivo. So we couldn't measure the affinity very easily. We tried lots of experiments that didn't work, but we did one kind of cheeky experiment to demonstrate that the drug binding sites that involve the interaction of the subunit, the green subunit, are actually the high affinity sites. We can see all the sites in the drug saturated structure, and they're all empty in the drug free structure. So what we did is take the drug saturated enzyme and then just wash it before freezing the grids to look at the drug occupancy. And our rationale was that if you wash the enzyme, you'd remove the loose bound drug. And when you do that, you can see that the drug density in the sites that involve this green subunit remains really quite robust, where the density in the sites that don't involve that subunit starts to fade. And what's more, you can look at the conformation of this one residue, phenylalanine 69, has a different conformation in the drug bound and drug free state. And you can see that in these two sites, the phenylalanine 69 residue adopts the drug-bound conformation. So that leads to this model of how bedaquiline actually inhibits the ATP synthase. You can see there's a very large conformational change that will occur as the drug binds to these sites that involve both the ring-forming subunit and the subunit that interacts with the ring. So there's one site on this side of the ring and the green subunit, and then that rotation of the ring is just of the right magnitude to produce an equal site, an equal size site on the other side of the ring that enables two drug molecules to bind and prevent ATP synthesis or hydrolysis. Now, one other important observation is that this drug bedaquiline seems to function as an uncoupler of respiration. So if you take a molecule like CCCP, which allows protons to flow across the lipid bilayer, you can induce respiration in mycobacteria. Bedaquiline also induces respiration, suggesting that it's allowing protons to flow across the membrane somehow. And it's doing that in a way that involves the ATP synthase, because if you make a mutation in the ATP synthase, you prevent that uncoupling. So that suggests one of two models. Either the ATP synthase is allowing bedaquiline to accumulate in the membrane and serve as an uncoupler, or something is happening to the ATP synthase when bedaquiline binds so that protons can flow through the membrane without making ATP and leading to this respiratory burst where the electron transport chain is working hard, but no ATP is being formed. And remarkably, when Gautier completed his titration of drug, he saw this uncoupling of the ATP synthase, where at low concentrations it prevents ATP hydrolysis, but as you increase the concentration of drug up to the micromolar range where the respiratory burst is observed, something seems to happen to the enzyme so that ATP hydrolysis can occur even though the membrane region is saturated with drug. So we're going to continue on this direction. There's a lot to be done looking at how TB drugs interact with mycobacterial respiratory enzymes like the ATP synthase. If we want to understand respiration in mycobacteria, we also have to understand it in eukaryotes because we don't want to inhibit it there. And of course, there's a lot to do with the vacuolar type ATPase and the development of new cryo-EM methods. So I've told you the names of the people whose work I've mentioned. I'll just end with this photograph of the group as it stood at the in the summer in a socially distanced picnic, and I'd be happy to take questions during the Q&A period.
Terrific. Thank you, John. So let's get us onto gallery mode. Okay. So thank you, everybody. We have just a couple of questions so far in the Q&A, so I encourage people to ask questions in the Q&A, or otherwise, I guess you can just speak up. If you have a question you don't want to put in the Q&A, you can just say, call on me in the Q&A, and I'll just call on you to ask your question. So the first question is to me, how do we know the impact of these different inhibitors, the P97 inhibitors, on protein interaction partners that may inform the differential functional outcomes? So yeah, this is something that's not as simple as we hoped it was. So we were hoping that these conformational blockers would then block certain protein-protein interactions, and we could predict that based on this up-down conformational equilibrium and have an answer to your question. But in fact, we don't really know exactly how that end domain conformation, remember this up-down conformation, how the end domain is coupled to the ATPase activities in the D2 domain, because some of the compounds that we would expect to have no effect on protein-protein interactions have a big effect, and then vice versa. So we're still trying to make that linkage between protein complex formation and ATPase activity. Now, the other thing is that we're looking at these isolated proteins expressed in E. coli with no post-translational modifications, no protein complexes that they exist in. So getting the context between, so that linkage between the structural biology and the cell biology is also a challenge. I think probably everybody on the call can, has experienced that kind of challenge. So maybe we can talk about that some more. To other questions. So for Lewis, any thoughts on the news on how DeepMind's AlphaFold performed at CASP? So maybe you can explain what DeepMind is and CASP, and then give your opinion.
Well, so I'm not an expert on much of this, but CASP is this competition that they have every year that I think John Molt at the University of Maryland sets up to see how well the predictors do with their prediction of structures. And I guess there's this new AI-based predictor that has really outperformed, and not by a little bit, but by a tremendous amount, sort of all of the competition. And it, at least about, I think it was 60 or 70% of the structures, it gets, at a level that you wouldn't be able to distinguish between experiment and prediction. So I think it's an amazing accomplishment for what it does. Now, it doesn't do so well with complexes. And how does it do with squishy proteins? I mean, I'm big advocate of protein dynamics and the fact that some proteins have multiple structures, and I'm not sure. I haven't gone through the test set to know how well it does with those more challenging situations, or really what the size of limitation is, and whether you can actually sort of recapitulate what the steps to the final product might be, so that you can make predictions about excited states of molecules, which I think is neglected, but very important as well. But anyways, it's a remarkable development, and I'd love to see, you know, all structural biologists be put out of business, but I don't think it's quite going to happen just yet. So,
well, this is what's in the ATOM consortium. We're also, the idea is that AI, machine learning, high performance computing will displace experiments, but of course it can't totally, because nothing exists until an experiment creates it, right? It just generates the hypothesis. So we'll have jobs for a while yet, I think. Anybody else have any knowledge or opinions about DeepMind and the prediction? Yeah, it's super interesting. Okay, for John, have you considered using a liquid cell to observe these reactions in TEM?
So that's an interesting question. I mean, in CryoEM, we really benefit. The fundamental limitation in CryoEM is the radiation damage of the protein. We would have been able to get to high resolution a long time ago if proteins were as radiation stable as gold or silicon. And we really benefit in CryoEM from the freezing process, which traps the reactive molecules generated during radiation damage of the proteins. And that trapping of movement of reactive species effectively gives us about 100-fold improvement, I believe that's the number, of the exposure of electrons that we can use. So for the equivalent resolution, we would have to find a way to do 100-fold better in terms of radiation damage, and it really would be extremely challenging to get high resolution insight in liquid. So I think for the features we want to observe at the resolutions we want to observe, liquid cell EM would be quite difficult. I never say never, but I think that's not currently practical with other limitations we have at the moment. I see there's another question also from Amanda about what resolutions we observe our proteins at. So current records for CryoEM are below 2 angstrom. People who are sitting there trying to push as hard as they can on a test specimen to see if they can get true atomic resolution. Usually the limitation for a biologically interesting specimen is the flexibility of the molecule, and it's much worse than that. So a lot of our samples are what we consider biologically interesting samples. We can do at around 3 angstroms or somewhat better. I should say that's probably a better 3 angstroms than you see in crystallography, because we are experimentally determining both amplitudes and phases. In crystallography, you're experimentally determining amplitudes and calculating phases. So our maps at 3 angstroms look really quite good to a crystallographer's eyes. And that's actually getting these high resolutions as part of the big challenges of consistently getting high resolution cryo-EM. We just discovered an issue with our microscope where it looks like we're losing some resolution in one direction. We probably wouldn't have noticed it for months if we'd waited to see the 3D maps. So that's really one of the challenges in EM right now, is making sure your pipeline is always giving you high resolution data at the resolution you need for getting the results you want.
Great, thank you, John. Okay, I'm going to ask everybody else's questions before my own. Lewis. Hi, Lewis. What gives rise, it's from Scott Prosser, what gives rise to slow dimerization of baby SOD? And what do you know about the cooperative processes of both the intramolecular events and dimerization?
Yeah, so the slow dimerization of baby sod, I mean, we think that it's basically just, you know, from the ground state of baby sod, and it's a thermal process. And it's a thermal process that basically leads to obviously a stable structure, since the dimeric structure is a very stable structure. Now, why do you get that structure and not others? You know, I think that structure is probably, again, just the stable one. So it tends to, there must be some built-in information, obviously, that says that, you know, there are certain amino acids that want to interact with other amino acids to form this dimeric structure. So there's a tendency to sort of force you in that direction. And in terms of the cooperativity, what do I know about cooperativity of the process? I actually think that the cooperativity is fairly small in the sense that you can get one but not the other. When you form the dimeric structure, of course, it doesn't have all of the features of the native dimer because it doesn't have the metal, it doesn't have the disulfide, and it also doesn't have the helix that is part of the loop, which then coordinates the zinc metal. And conversely, of course, when you do get the second process, the transition towards forming that helix that can coordinate the metal, you don't have a dimer formed. So I think that these are kind of like one-off processes. I mean, they're very rare events, or quite rare events, in the sense that the population is small, and they're also short-lived. So it's not surprising, given the low probability of any one of these processes happening, that you don't have you don't have both happening in a single event.
Great, thank you. Jason, are there representative refolding mechanisms within the repertoire of heat shock proteins?
I'm going to point to a paper by Lewis K, I think. There's a couple of different ways you can think about mechanisms for refolding and the interaction with molecular chaperones. So you could think about molecular chaperones, knowing what the folded state is, right, and guiding proteins to that folded state. That doesn't make a lot of sense, right? Because then you need a specific molecular chaperone for every single type of fold, and that doesn't seem to be the way the architecture works out. So instead they use more general type mechanisms, and I think some of the subset of general mechanisms are things like maybe the chaperone binds to a misfolded state and dissolves it, or in other ways reverts it to the unfolded states and gives it another chance. But I think in all seriousness, I think Lewis's paper on DNA-K from a couple of years ago it was really clear, and I think it's similar to work from Jim Bardwell at Michigan has shown similar things for other chaperones, and I think this is starting to become probably the answer to this question is instead chaperones really just bind to the unfolded state and not the folded state. And so for things like heat shock protein 70 substrate binding domain binds to a stretch of any 7 or 8 uncharged amino acids, so hydrophobic or neutral. And so those types of residues are often found on the interior of a globular protein. And so in the unfolded state, the molecular chaperone can bind them, but then when the protein folds, it hides those states on the interior, and now there's nothing for the chaperone to bind to. So by selectively binding to the unfolded state and not the folded state, you slow down folding. Actually, the molecular chaperones in the lab all the time, they dramatically slow down folding, but they decrease aggregation and other off pathway things through that same mechanism. And honestly, I think, Lewis, your paper on DNA-K, which we talk about all the time in my lab, is probably the cleanest example of that. I'd be interested in your thoughts.
I agree. I think it is the cleanest. No, I think you said it better than I could. I mean, it's really, I think it does, look for unfolded regions, and it actually just, holds onto them and gives the protein another kick to be able to, it binds, it comes off. There's multiple binding sites, so it's almost like you can reset. If it binds to a certain region, and then you can get secondary structure that forms, you don't get the ternary structure until this thing comes off, and then it tries to fold. If it doesn't work, it binds again, but the likelihood that it makes the same mistake as the first time is small, because there are so many different binding regions that it can bind to a different region, and then it just sort of resets the timer, and maybe that's a better region, and then it tries that and keeps trying until it succeeds, or the proteasome decides to do its thing, runs out of patience.
So another way I think about the chaperones, and tell me how general or how true or untrue this is that they're a regulatory mechanism. You want to keep the protein in an inactive state. You hold it in this unfolded state until the ligand comes in or the context is right.
Yeah, and I think the steroid hormone receptors, of course, are the classic example of that from Pratt and Toft and Smith and more recently David Agard and others, right? So the chaperones definitely hold the steroid hormone receptors in a soluble, importantly, really soluble and capable of binding to their ligand, but not folded, right? And on Lewis's energy diagram, they sort of trap the system at somewhere, not quite the native state, but somewhere else, and they specifically hold it there. And that, I would call that regulatory, right? And then in response to ligand, and then of course the delta G of the ligand binding, of course, changes where you are in that diagram, the chaperones release, and then you proceed down the pathway. I do think there's a lot of examples that I think is probably not the only way that they work, right? But I think in terms of like taking things across membranes, you want to be able to hold proteins in an unfolded state so they can translate through Sec61 or through the Tim-Tom complex in mitochondria. That's, there's certain architectural topological constraints of those transporters. And so I think for those subset and things like steroid hormone receptor, that's going to be really, really important. Other proteins, I'm pretty sure, interact with the chaperone very little, maybe when they're first born, and then that's it. The protein's folded and it goes about its business and probably doesn't see the chaperone ever again. So
yeah, it's interesting how nature makes use of the same kinds of things in lots of different contexts. Okay, so question for me. I'm going to answer the second one first because I can answer it. So on a separate note, what do you think is needed to advance fragment-based drug discovery? So accuracy versus high throughput. Yeah, so what I like about biophysics is that it's a more the merrier kind of thing. That every time you do an experiment, you learn something, you don't learn something else. And so really looking at a series of orthogonal experiments is the way for accuracy. Of course, that can also reduce the throughput. So the way we think about it is that if the good hit needs to do A and B and C, do whichever one is faster first, because it has to do all three of them eventually. So you get your throughput by using a funnel-based approach. If hopefully that makes sense to Scott, and feel free to ask again if I didn't answer your question well. I think the real challenge in fragment-based drug discovery is fragment optimization. I think we're really good at discovering fragments. Lots of different techniques have worked and they continue to be refined. It's developing those pipelines that really still the state of the art in fragment-based optimization is structural biology. So anything that speeds up structural biology will speed up fragment optimization, but also technologies, computational approaches, other lower resolution technologies that give you site-directed information to allow you to do better experimentation, better chemistry, faster chemistry is going to really move that field forward. Any other, Jason, anything you want to add to that as a fellow medicinal chemist?
Drug discovery is hard.
Drug discovery is hard.
That's what I've been looking for easy ways of doing it for a long time now. I've yet to find one.
It's true. Actually, that reminds me of a question I want to ask John. So when you have that compound that binds in 3 different sort of related sites, do you think that you need to bind both? Like you had the 2 compounds kind of binding on opposite sides of this rotor. Do you need both to stop it in both directions, or do you think either one?
Yeah.
Work?
So actually, the building on the drug discovery is hard. I'd add drug discovery is really cool because the drug discovery process there selected for a molecule that's just the optimal length to maintain the contacts with the rotating part and position the other parts in 2 different sites in just the right position. So it's almost like evolution in that it found the optimal solution to the problem. Now, that suggests that it is important for it to bind 2 sites. Although we think that one of the 2 sites is the higher affinity site because it's deeper, it has more van der Waals contacts, it explains the structure-activity relationship of a bromine group that is essential for the drug to be active. We think one is more important, but the fact that it's just the right size of drug to fit those 2 sites simultaneously suggests that it didn't evolve there for a purpose, but it was selected to do that for a purpose.
Yeah. So I appreciate that. And that's when I think about phenotypic drug discovery, that's what I frequently am amazed by is what unusual mechanisms a compound can do that would just be very difficult to predict. So you want to give it a lot of opportunities to do something useful and then figure out how it does it. And you get a lot of it there. Okay, the other question for me, and maybe actually the others have better answers than I do. What are your thoughts on optimal computational methods to evaluate allosteric pathways? Yeah, So I always really, I mean, it's old now, but I always really like this idea of looking evolutionarily at residues that are co-altered during evolution and seeing if you can't build networks just based on mutual exclusivity and things that seem to be distant from each other but change in parallel. I know there are a few companies that are working on this area, and so they've been pretty mum about exactly how they're doing it. Does anybody have any insights, recent stuff they've seen on allosteric networks? No, it's a real area of, it's a really interesting area. I mean, even empirically looking at networks is challenging enough.
I mean, you just sort of think about simple experiments that we often do by NMR, and that's it. You make a mutation. You make a mutation someplace and you see what happens, right? So that's kind of a stupid experiment in the sense that you're just sort of blowing up a part of a protein and see, where the fallout is. But you can, you may be able to follow things along. I mean, that's essentially what we do experimentally, but you can do it in silico as well. I mean, you make a mutation and you see in NMR, what the changes in chemical shifts are. And then you correlate that change that you've made functionally. So that can give you information about how things traverse from one region of the molecule to another.
So then I imagine molecular dynamics allows you to do the same thing. So you say, okay, this is the dynamics of the system. You alter the system and you see what the changes in dynamics are. I just don't know how well those have, I just haven't looked to see how well those really translate into making decisions about allosteric networks. The other thing, along this line of making mutations, I really like the idea of site-directed fragment discovery or site-directed drug discovery for this approach. So then you cram something in a hole that you think is transiently there, then you force it to stay there, and see how that mucks up the system. There's been some nice work on beta-lactamase around that in particular.
Michelle, I think one of the other interesting things there, maybe, on Lewis's mutational idea is, deep mutational scanning, which we're starting to try to get into, and with CRISPR, being able to make thousands and thousands and thousands of mutants, like you used to be able to do in yeast, but now, you can do in mammalian cells. That's, I know the question was about computational, but I think being able to very, very quickly do deep mutational scanning on a target to really understand those non-obvious things where you have an amino acid switch way out on the surface somewhere, and yet, in a phenotypic readout, it clearly is changing the outcomes. I think that's going to be a powerful complement to the strictly computational approaches too for finding or mapping allosteric sites and maybe druggable allosteric sites.
Mm-hmm, yeah, and networks too, how. Great. So that's all the questions that we had in the Q&A. Still feel free to add any if people on the line have questions. And then how about each other? Any questions for each other?
Could I ask one?
Definitely.
So I'm going to ask a P97 question, as you might have guessed. So I'm just interested, Michelle. I mean, you showed that P47, probably 37, like to bind in the upstate. So, and there are other proteins that like to bind in the downstate. And I'm quite interested in that. And I wonder, what's your take as to why that's the case? I mean, we have some ideas, but why do some like the up and some like the down? I mean, the molecular level, what do you think is going on there?
Yeah, well, at least in the UBXD1, which I know you know very well, it binds to other sites on the stalk of the protein. So there's this composite binding site that is closer in the downstate. So a postdoc who, after Stacy, a professor now named by the name of Mbika Bagi, her just sort of basal hypothesis was oligomeric proteins are going to prefer the upstate because then the proteins are closer to each other. So the binding site is kind of on this downside here. So it just brings them closer to each other. And the monomeric proteins wouldn't care, except for the UBXD1 or other things like it that have secondary sites along the stalk or at the C-terminus of the protein. And she did this for 7 or 8 proteins, and it basically held true. Challenge is that, as Lewis has demonstrated, the energetic differences between the up and the downstate are really small. So even this pretty weak mutations that don't alter the binding of that by NMR, just they don't dramatically stabilize the upstate or dramatically destabilize the downstate, I guess is the way I would predict it. They still have an up-like conformation because you've got 10 nanomolar or 25 nanomolar protein-protein interaction. I guess it just pulls it up. But this is something I've always wanted John to tell us, is when you're in the low affinity state, are you pulling up the protein or are you distorting the adapter protein? Because I think the mechanisms of what that means in biology might be different if it's crisply one or the other.
Lewis has probably thought about this much more than I have. I'll pass that to Lewis.
Yeah, so it's quite, we did a study and John was involved in some of the CryoEM as well, where we looked at P47. And P47 is a wonderful molecule for NMR because it has large regions of intrinsic disorder that really can't be addressed with other techniques. And what we were able to identify is, in addition to, so they have these various ship domains, the C ship, but we noticed, we realized that there's an additional ship domain that was just sort of staring us in the face. And then in the upstate, what can happen is that, so there's like different prongs on P47, let me put it that way. Two of the prongs combine to a single arm of P97, but then an additional prong on P47, this sort of new ship domain combined to an adjacent prong in the upstate. And apparently can do that for reasons that I don't understand much more readily than in the downstate. So you actually have an energetic component which says, hey, I would rather interact in the upstate. Eventually you can compete off that if you add enough P47 molecules, they'll all want to bind, and therefore they'll kick off that additional interaction with the adjacent arms, so that all arms can bind is separately individual P47 molecules. But it's interesting that these sort of, they're kind of like super adapter molecules, and that they have a lot of different sites that can be exploited. And I wonder if it's just that sites reaching in various ways that are easier achieved in an upstate or a downstate than vice versa. And again, we're talking about subtle energetics for sure, but so you don't need very much to flip the switch, which is why I always thought that P47 should be a wonderful molecule to drug, because you don't need very much free energy. And you don't want to put much free energy in the system because you don't want to lock it up, or you don't want to lock it down because it does so many different functions, right? You've got to have it be able to readily adapt to a world that's complicated.
Yeah, it's true. So when we just do the easy experiment, the ATPase experiment, we see inhibitors of ATPase activity, obviously, and then they're far away from the conformational change. So we're working on other approaches to really focus on the up-down conformation. So what in the D1, in the ND1 domains alter this up-down equilibrium? And the first experiment that we did a long time ago now, we ran an NMR fragment screen and on the N domain, on the protein-protein interaction domain. And wouldn't you know it, none of the fragments bound at the interface with all for people who don't know, the N domain has got these two lobes, and the space between the lobes is where most protein bind. And some proteins are long alpha helices and some are globular domains called UBX domains. And they bind in more or less the same place. All of our fragments bound next to it, like one helix over in this little binding site. And they tended to stabilize the protein-protein interaction. So I think they just firmed up a conformation that stabilized the interactions. And we were trying to figure out what to do with it. And then a paper came out showing that ship domain peptides that Lewis was talking about bind in that site. But then we couldn't really get them to work. We need to do some other stuff to get those to work as peptides. They didn't bind tightly enough to really displace the fragments and so forth to really validate that. But just to show you that there's these high affinity sites that you see easily structurally, and then there are these lower affinity sites that molecules can bind or peptides can bind. And when put in this oligomeric context in particular, they can be really important for biology, but difficult to study biophysically separated. I think, Jason, that's a real theme in Jason's work too, right? These low affinity rapid sites still having a huge impact in biology. Any other questions, science questions for each other? A new question just came in. Let me see if it's for Jason. Is higher binding affinity always good for drug development targeting protein-protein interactions? What if off rate is important so as not to drive passengers down pathways, using the London Underground technology analogy, that could be detrimental for the cell or the organism as a whole? So I guess a dead end complex idea.
Yeah, it's a really, really good question. I try to ignore that question all the time because if I think about it too hard, it makes the job too hard. But it's true. It's really true. You know, what is the sort of Goldilocks sweet spot for the affinity for drug development, especially on these types of targets? And you know, one can think about the you know, really the dwell time is really what's going to be important, that sort of balance between that on rate and the off rate, and the amount of time that the small molecule is bound to the individual target. And I think it's good to always take that question and break it into two parts, because drug discovery is a little bit different, right? Because drug discovery, you need to be able to deliver a small amount of the molecule. It needs to bind very few other things in the cell, can't be toxic, and needs to engage with the target. You know, that needs, you have pharmacology and pharmacodynamics going around with that activity of that small molecule on that target in a living animal, in a human, in a patient. Chemical probes are a little different. If you're trying to ask a question about the importance of that particular node in the London Underground, right, then really you want to just shut that off entirely to sort of reveal the role of that particular node. So I think for chemical biology, which we tend to start with, you really do want to just drive down on affinity as long as you have selectivity. That might not be the best strategy for drug discovery because you might get long-term tox, but I think, you know, in an academic environment like ours, I think starting out with chemical probes being the goal is probably the right way to do it. And that's, or at least, let me change that. That's how I like to approach that problem. And then if it looks like there's a there, then you can sort of go back and look through your MedChem series and pick out things that might be better translational leads. So it's a good question. And I think I'd like to think about it in sort of two layers. I know you think about this too, Michelle. What are your thoughts?
Yeah, I'd say the same thing. Adam Renzel is a medicinal chemist that we work with. He describes them as drug prototypes. Yeah. So we start with chemical biology, and chemical biology, actually, if you're going to use it as a tool, it has to be a much sharper scalpel than a drug does. Drug just has to not be toxic. It can hit all kinds of things, but you'll make the wrong decisions if you have a chemical probe that's not specific. So in a way, you come up with better, maybe they're more potent and selective than necessary for the drug, but usually they don't have the in vivo properties and so forth that we need. Yeah, I think of it the same way Jason does.
Actually, so for Michelle and Jason. So it seems to me when you think about drug discovery, the prospect of doing massive screens with thousands or hundreds of thousands of compounds is extraordinarily daunting. And what are the prospects for methods where either structurally by CryoEM or NMR or with mass spec, you just take your known target and subject it to thousands of things in one pot and then try to figure out what it's interacting with? Is there any progress on that front in either mass spec or like solving the CryoEM structure to figure out what bound after mixing?
Yeah, people do that. Yeah, people do it with crystallography in pools, not thousands, tend to be pools of 10 or 20. What you end up with is a solubility problem. It's completely boring problem that plagues chemistry is, you know, you just can't get 1,000 things to all be soluble at the concentration that you expect them to bind. But pools, definitely, there's a technology a lot of people like called ASMS, affinity selection mass spectrometry, where you basically flow a molecule down, and if it binds to something, it's slowed down, or if it binds to something, you can isolate it based on binding to it.
Has that led to any useful compounds or any useful hits?
Usually it's used as a secondary screen because actually screening hundreds of thousands of compounds for function is not so hard. It's screening for binding that's harder to do and super high throughput. Yeah, the differential scanning fluorimetry is a plate-based technique that's pretty fast. The surface plasmon resonance is pretty fast. NMR is also in pools pretty fast, but usually not as fast as function.
And I think, you know, John, the thing I totally agree with all that. And the thing I would sort of add is that I think, the early phases of drug discovery now that you're talking about, really finding scaffolds that interact with your target, that's becoming, I think a little more holistic than it used to be, where you just took your target and screened blindly against whatever 10 million compounds from AbbVie's deck or whatever. I think it's becoming a little bit more of a hybrid situation where you do some of that still, because like Michelle said, it's not that hard to screen 150,000 molecules or 250,000 molecules. That's not that hard. But you sort of pick which molecules you're going to do with some computational and some structural and some modeling and some docking, and maybe picking subsets of libraries that you think you have some reason to think are going to be involved. So I think it's blindly going in and just screening 10 million molecules. I don't think that happens very much anymore, to be honest. I think it's more of a, you take your target, what you happen to know about it, take a couple of different approaches, including screening hundreds of thousands of molecules, but buoyed by other types of approaches, and especially computational, and then holistically coming up with your scaffold choices. Do you agree with that, Michelle?
Yeah, definitely. And it really depends on where you're going. If you're starting from phenotypic, or you could think of it as reverse and forward chemical genetics. If you're starting with your target or with your phenotype. And what you see, I think you see this a lot in pharma too, is that they have these big libraries, but they'll go in with a smaller library that they can then afford to do much more complicated biology on. So the biology version of your question about high throughput biophysics would be high content imaging microscopy, organoids, much more complicated primary cells. And so we do this in our lab too, and we see this in the industry generally, that smaller, more complex libraries, more iterative testing of compounds. And because I think it's too tempting to do something that's really fast and easy, but is not relevant because you've simplified something important away by accident, like a domain instead of a protein, or protein without PTMs instead of cell context. Yeah. So along those lines, what do you guys think about the prospects for chemical optimization using CryoEM?
I think it's inevitable.
It's inevitable. Thank you. You heard it here.
It's CryoEM is not as fast as crystallography, but it's getting faster and faster very quickly. I mean, it used to be a PhD thesis to get a structure at 20 angstroms resolution, and now it can be a matter of a week. So we need to get it down to hours, or not necessarily but somebody has to get down to hours for it to be really useful for pharma. But I think it's inevitable.
Yeah, and the other question that you hear, so you have this concern about speed. And again, I agree with you. It seems like technology, that's a technology problem, and that will get solved, right? That's an engineering problem. But the resolution, you mentioned that a 3 angstrom resolution is better than a crystal structure at 3 angstrom resolution because you calculate it differently.
Sure.
For small molecule optimization, resolution's good enough?
You really want around 2 angstroms, I think, there to see a methyl group or carboxyl or whatever. But it'll come. I mean, the resolutions are each year consistently, the standard is changing for what is a good map. And so I think there's just the regular, kind of boring regular advances and everything getting tighter and better, and we'll get those resolutions routinely.
Yeah, I think that's interesting too, because a lot of times, Jason was talking about merging with computation. It's the same. We know where, once you know more or less where the compound binds, it's not that complex a computational approach to say, drugs tend not to be very flexible. So I can't see the methyl group, but if I can see the ring and I know where the methyl group is, I can.
And for biologics, I mean, 15 angstroms is enough to figure out where the epitope is. So it really depends on the question you're asking. But for, I think, medicinal chemistry, it was still a little bit off. To do it routinely can certainly be done occasionally now.
Yeah.
Can I ask a question along that too? I think as Lewis knows, fluorine and NMR is incredibly important in drug discovery, right? So changing the chemistry to install fluorine is super powerful in drug discovery. And cryo-EM is about electron density. So is there any play there about sort of changing, putting like lanthanide series metals or something with a really high electron density to help that process?
So Scott Prosser, who asked all the question, many of the questions, is a fluorine NMR aficionado. So cryo-EM is not about electron density. Cryo-EM is about integrated Coulomb potential. X-ray crystallography is about electron density. And integrated Coulomb potential goes up with Z, not Z squared. And so you don't get the gain that you'd expect by using heavy atoms.
Yeah.
But you can see them a bit better, but not, they don't blow up in a map like they would in crystallography.
Cool. Thanks, John.
Maybe I
Okay.
can just,
Yes, go
Right.
ahead. I just responded to your chat
okay.
to John by accident. Yeah, go ahead.
Maybe I can just use the last few minutes to ask you guys about our Global Challenges program and what you guys can gain. I mean, clearly, you guys have a lot to talk about, and common interests, I think different ways of coming at these really interesting problems. Can we find a way to make this program benefit your collaborations and interactions even further? Can you see a benefit in sending people back and forth and identifying some goals to work towards collaboratively? I know some of you are working together already, but give me your thoughts about that.
I mean, personally, I'm all ready to send graduate students up to Toronto to look at some of these, look at ATPases, AAAs by Cryo and by NMR. Lewis and I have talked about collaborating, sending compounds back and forth for a while. So I think it would be, I think students would really enjoy it, especially now, once you can start traveling again, it's just gonna be like loosed into the world. Because there's something about being on site that we can send compounds back and forth, but there's nothing like being there to really learn the technologies and bring them, and the ways of thinking about a problem and bringing them back to the lab.
Yeah. Great.
Yeah.
Okay.
Anybody else? Yeah, want to?
Yeah, as somebody who thinks about spin Hamiltonians more than he thinks about proteins, I'm always looking for collaborators. I should put up a big sign looking for collaborators. So I would very much welcome people who would like to think about using NMR in preferably ways that have never been used before, because if they've been used before, there are probably better people than I to work with, but to sort of do the next experiment.
Yeah, I think for CryoEM, we've talked for the 20 something years I've been in the field that eventually would get the resolutions where you could start to think about proteins and drugs, and suddenly we're here and we are thinking about it. So and we don't know anything about it. So finding relevant collaborations for the systems we're interested in, where collaborators have expertise in drug discovery, is certainly of interest. And we're really babies in that area, so it's quite exciting.
So if I may, Shanna, so Jason, I guess we could say that Lewis and John are more technique people and you're more of a problem person, sort of divide the world that way. How do you choose collaborations? How do you know when to start a collaboration?
Yeah, I often say a similar thing. I'll paraphrase Lewis, but in my own way. If there's a really trivial drug target, like a kinase or something like that, not that interested. I like hard problems, hard drug discovery problems. And so, I'm always sort of looking out for something like that, something that's got a really interesting technology that cracks open an old target or a new target that's really difficult that pharma's not going to touch, right? And you need some sort of trick or some element of patience that they don't bring to bear on the question to be able to solve it. And I think that's sort of the sweet spot of what we do. Maybe you too, Michelle, in some ways, right? So we go after this sort of more challenging type of targets.
Yeah, I would say that we like the targets that require technologies to development or reuse of technologies to crack the problem. And so then when you do have those technologies, then you do some collaborations where people have a target that your technology is well suited for, and then also you have a problem and somebody else's technology is well suited. So it's there are lots of points of intersection.
One comment, Shana, on your question too is I think oftentimes these things work good in sort of the organic sort of grassroots type way, honestly, right? So there's a lot of tools you can probably spend a lot of money on to do project management and map out how things should connect between UCSF and Toronto. I don't know that actually fits human nature. I think things like this are great, which is why I was so excited, that you put it together. And I think just, and I'm glad that there, presumably there's a lot of trainees in that participant list, right? And the young people with really cool, good ideas of how they want their careers to go. And then they can sort of listen and make connections and build things organically. And I think that matches my version of how science works and how people work, and how creative people work best together. And the really, really, really key role for junior people, coming in and generating new things that I don't see and John doesn't see, and they'll see some connection between things. I think that's what's important.
Yeah.
Okay, well, it's a good segue into what we're going to talk about tomorrow, which are high throughput technologies for biological discovery. And I think we should keep our eye on some of the screening type tools that people will talk about tomorrow, because they may be those types of approaches that can kind of crack open a wonderfully challenging problem that then we can, hand to you all. So in any event, this was a great session. This was exactly what we hoped for, to get people from either side, with these shared interests. And clearly there's lots to continue discussing. So I'll ask you guys, keep in mind, we're, going to keep developing this and obviously launching it once people can go back and forth. But I completely agree. It should absolutely be grassroots, no software involved. There's definitely plenty of points of connectivity. So I think with that, we should wrap up. And I just remind everybody who's stuck with us that we'll be back tomorrow. So same time, we will have 4 talks that are more kind of on the technology side, high throughput biology, but very complementary to the kinds of things that we heard about today. So please join us if you can. I want to thank all of our speakers. It was just unbelievable science and such a pleasure to hear about it. So thank you so much for your time. Please, let's all keep thinking about this, and then we'll circle back as our ideas kind of continue to advance, and then when we're ready to roll out. So thanks again. Take care, and I'll talk to you soon.
Great. Thank you, everybody. See you tomorrow.
Bye.
Bye.
Bye-bye.